Dr. P. Lakshmi, postdoctoral fellows, research scholars, students, and supporting staff. I take this opportunity to welcome you all on behalf of the Human Genetics Department to the inaugural function of the international webinar organized by department in connection with the superannuation of Dr. V. Lakshmi Kalpana. I extend a hearty welcome to the chief guest of today's function, Dr. P. Shyam Prasad Garu, Honorable Vice Chancellor, Dr. NTR University of Health Sciences, Vijayawada. I extend a warm welcome to the resource persons who readily accepted our invitation, Dr. R.V.R. Prasad, Dr. Raghunatha Rao Garu, and Professor Mitashri Mitra. I welcome all the participants of this webinar, comprising research scholars, faculty, doctors, and press and electronic media. Let us start the program. I request respected Principal, College of Science and Technology, Professor Srinivasa Rao Garu, onto the dais. I request the class representatives to honor our principal. Now we'll start the program with the prayer song.
ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ಪಾಶಾಂಕುಶಧರ ಮೂಷಿಕವಾಹನ ಸರ್ಪಾಲಂಕೃತನೇ ಗಜಾನನ ಪಾಶಾಂಕುಶಧರ ಮೂಷಿಕವಾಹನ ಸರ್ಪಾಲಂಕೃತನೇ ಗಜಾನನ ಆದಿಪೂಜಿತ ನೀನು ವೇದವಂದಿತ ನೀನು ಆದಿಪೂಜಿತ ನೀನು ವೇದವಂದಿತ ನೀನು ಮೋದಕ ಪ್ರಿಯ ನೀನು ಗಜಾನನ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು.
Dear students, now we'll have our traditional lamp lighting ceremony. I request our principal sir to light the lamp. I request Professor Sudhakar, Dr. Lakshmi Kalpana Madam, and Honorable Professor Paday Garu to join. The Department of Human Genetics, first of its kind, was established in the year 1972. The department offers Master of Science program and PhD program in human genetics. The Human Genetics Department has well-equipped laboratories in the areas of biochemical genetics, cytogenetics, molecular genetics. The department also has well-organized computational laboratory for bioinformatics with 24 hours internet facility to the students. Presently, the faculty strength is 6, and there are about 70 MSc students and 16 PhD scholars, 4 postdoctoral fellows. Alumni of this department are well settled in research organizations and laboratories at national and global level, and in hospitals as genetic counselors. The department offers diagnosis of sickle cell disease to the patients who mostly come from the agency areas like Paderu, Araku, Kukunpeta, etc. The department also has MOUs with several hospitals for research-oriented studies. Several projects from UGC, DPT, and ICMR have been successfully completed, and currently there are 3 ongoing ICMR projects. Several papers were published in various national and international journals. The department has conducted several national and international workshops, conferences, and seminars where the faculty and students had the opportunity to update their scientific knowledge. As a part of this, the department is conducting today's international webinar on recent advances in human genetics in connection with the superannuation of Dr. V. Lakshmi, BOS chairperson, who served this department from 2002 in various capacities. The theme of today's webinar is Recent Advances in Human Genetics. It provides a platform for dissemination of the most fascinating and significant contributions in the field of genetics. Human genetics is a fast-growing field where enormous progress has occurred over the past few decades. Advances in genome sequencing and structural genomics have opened up a unique opportunity to better understand human diseases at the molecular level. One of the most revolutionary advances in this new era, cell and gene therapy. In recent years, the researchers have taken the advancement one step further with genomic tests of different diseases cancers. The recent advances in human genetics can provide a more precise diagnosis of diseases with a personalized treatment strategy. The progress made in from rapidly developing field is outlined in this webinar, focusing on the latest advances in cell and gene therapeutics, cancer genomics, and precision medicine. So with this opening remarks, now I request Honorable Vice Chancellor, NTR University of Health Sciences, Dr. P. Shyam Prasad, Chief Guest of today's function, to give the inaugural speech.
Thank you, Madam. Am I audible?
Yes, sir. Yes, sir. You're audible.
Respected Professor PVGD Prasad Reddy Garu, the Chief Patron, Vice Chancellor of Andhra University. Other patrons include Professor K. Samantha, Rector, Andhra University, personally known to me, Srinivas Rao Garu, Principal, and Krishna Mohan, Registrar. He is my teacher in my MBA. Director Sudhakar, Lakshmi Kalpana Garu, Lakshmi Garu, Department of Organizing Members in Human Genetics like Uday Kumar, Mahankali, Sridevi, Rajeshwari, Lakshmi, Sudhakar, Vindhya, and today research persons like Venkat Rattamala, my good friend Raghunath Rao, Maitrishi, Mitra, and good wishes for international webinar on recent advances human genetics, RAHC 2021. I am from the Department of Genetics, and I know both the human genetics and medicine has intervened in so many things. The basic genetics scientists have made medicine easy and prophylactic, mostly preventive aspects also seen nowadays. And there are certain people I still remember when I was doing my MCH pediatric surgery at KGH Andhra Medical College. I suppose Satyan Narayan Garu was the head of the Department of Genetics. He used to help us in karyotyping, see various genetically based diseases, like we have seen so many things like cleft lip palate, cancer, and other things. He helped us so much genetic counseling at that time. Last 4 decades has entirely revolutionized the Department of Science, Genetics, medicine. The approach we see where genetics helps us medicine in a very big way. Things like that is already mentioned, where there is a chronic obstructive pulmonary disease, a disease of pulmonology, as inevitably making so many problems in the world, and they become interpolated. After 2030, it is one of the important non-communicable diseases. Way back 40 years back, we have seen infectious diseases like diarrhea, dysentery, children, fever, and other things. They are all disappeared nowadays. We have only non-communicable diseases, out of which the big things come back with a cancer, diabetes, hypertension, and other disorders. So it constitutes 90% of the diseases in the world. So we have to focus more on the non-communicable diseases like heart attacks, trauma, like that. And we know certain diseases which have a basic genetic background. How do you prevent this disease? You can identify this is the thing which is responsible. For example, they have 2 genes isolated from carcinoma of breast, what is called BRCA1 and BRCA2 and herpesipin. If you identify, you remember in the literature, there is one actress in American scenario, Angelina Lewis. she had identified as BRCA1 and BRCA2, where they are suffering with or they are able to get a disease called cancer breast. Next 40 years, she has gone subjected to the percutaneous bilateral mastectomy and implantations. So she is the woman, she utilized the genetic knowledge in the world and she has gone to the surgery level in a very big way. So what we suggest is this genetic knowledge should go in a very closely moving with medicine, and we can sometimes prevent a disease. If you say a genital counseling is more important for us and you are telling us new things for us, cancer, genomic things, and you identify so many things. In carcinoma breast also, we are not only BRCA1 and BRCA2, there are about 8 genes were isolated and each one have a hereditary component, and what way, how it is spreading to the common man is very, very important. The statistical data says in India, we have a 1,80,000 people are affected in 2018, out of which 75,000 people are dying with carcinoma breast. What is the best way to identify the disease? Very early stage, it is from clinical aspect. From prevention aspect, we should go back to the genetics. Ask you people to find out how best we can identify as early as in the life. Go for a mastectomy, or if people are recessive gene, don't marry. Any expression can occur in the second generation or third generation. So how to prevent a disease? Disease can be prevented number of ways, primary, secondary, tertiary, like that. So the basic genetic structure should be studied for all diseases probable. I'm in the department of seeing their work. I was really amazed. The work of Sudhakar and his team have done great work for last 4 or 5 decades, right? And they keep abreasting with the national and international symposiums. And I have personally visited Paderu to see their work in sickle cell disease. And I was informed that he is one of the principal investigators in management of sickle cell disease in Andhra Pradesh. I see so many people like that, and I wish you a grand success of this international webinar in genetics, useful for the young generation. They have an impetus to grow enormously in future. I wish you good luck, happy, very happy things, sir. And I thank the authorities to give me as to share your views in this particular. I try to observe or hear your symposium from Vijayawada. Thank you very much, sir. Namaste. Sai Ram, sir.
Thank you very much, sir. You have enlightened us about the different kinds of diseases that are there, including the lifestyle diseases. You have mentioned the different types of cancers, which are hereditary, and the genes that have been identified in causing cancer. Thank you for your best wishes, sir.
Welcome, madam.
Now I invite our respected principal, Professor K. Srinivasa Rao Garu, to deliver a message on this occasion.
Honorable Vice Chancellor of Andhra University, Professor PVGD Prasad Reddy Garu, beloved Rector of this university, Professor Samatha Garu, very dynamic and able administrator, Professor Krishna Mohan, the Registrar of Andhra University, and today's chief guest, Honorable Vice Chancellor of NTR University, Health University, Professor Shyam Prasad Garu, Dr. Lakshmi Garu, Professor Sudhakar, Professor Padaya, Dr. Lakshmi Kalpana, young dynamic contract faculty of this department, research scholars, and my dear student friends. Very good morning to you all. It's a pleasure for me to participate in this international webinar on recent advances in human genetics, which is conducted in conjunction with the superannuation of an eminent scientist and teacher of this department, Dr. Lakshmi Kalpana Garu. I am very happy. This type of felicitations in honor of a particular faculty when they superannuate conducting is a very good gesture, and this will motivate all other people, other faculty, as well as the young students to continue their good research work. In this direction, I congratulate all the faculty members, especially Professor Sudhakar, Dr. Lakshmi Garu, and her team for organizing such a webinar. And the theme is also more relevant to the present day needs of the society, because human genetics is more associated with the public. The geneticists evaluate, diagnose, manage patients with hereditary conditions or congenital malformation, genetic risk calculations, and mutation analysis, as well as refer patients to other medical specialists. The geneticist carries out studies, tests, counsels patients with genetic disorder. In recent years, much more has been learned about human genetics through chromosomal analysis, biochemical studies, and because of this, facilities for genetic counseling are assuming important dimensions in preventive medicine. As the general public become increasingly aware of prospects and possibilities of advances in this area of medicine, it is likely that the scientists will be approached by patients seeking help and support concerning matters related to genetics. Extraordinarily, technical advances in the field of human genetics over the past few years have catalyzed an explosion of new information about the genetics of human autoimmunity. In particular, the ability to scan the entire genome for common polymorphisms that associate with disease has led to the identification of numerous new risk genes involved in autoimmune phenotypes. Several themes are emerging in this direction. Autoimmune disorders have a complex genetic basis. Multiple genes contribute to disease risk, each with generally modest effects independently. In addition, it is now clear that common genes underlie multiple autoimmune disorders. There is also heterogeneity among subphenotypes within a disease and across major racial groups. Array comparative genomics hybridization, usually known as array CGH, is a method used to detect segmented DNA copy number alterations. Recently, advances in this technology have enabled high-resolution examination for identifying genetic alterations and copy number variations on the genome-wide scale. The current genomic array platforms and CGS methodologies highlights their applications for studying cancer genetics, constitutional diseases, and human variation and disease visualization, and analytical software programs for computational interpretation of array CGH data. Another important part which comes in the human genetics research area is obesity. Obesity is a heritable trait that contributes to substantial global morbidity and mortality. The genetics of general adiposity, a measured by body mass index and abdominal obesity as measured by waist to hip ratio have distinct biological backgrounds. Gene expression associated with general adiposity is enriched in nervous system. In contrast, genes associated with abdominal adiposity function in adipose tissue discovers emerging from population genomics are identifying new disease markers and potential novel drug targets to better define and combat obesity and related diseases. As an era of targeted genetic testing gives way to global mainstreaming of high output genomic sequencing, important decisions need to be made regarding which genetic variants associated with conditions are considered worth knowing. In addition to the strength of genotype-phenotype correlations, the ability of effective treatment or prevention and significance of the genetic findings to wider family members, the seriousness of the conditions has been pivotal to policy decisions governing the use of genetic screening. Some topics in present-day research are the impact of expanded carrier screening, direct-to-consumer genetic testing, widening of the presumed anonymity of genetic donors by advanced genetic testing, advances in the research of genetic causes, underlying male and female infertility, utilizing massively parallel sequencing in pre-implementation gene testing and non-invasive parental screening, mitochondrial replacement in human oocytes and other areas. I hope this current webinar focuses on these recent discoveries within the functional related groups of genes that provide close to novel pathways of human genetics research. Our College of Science and Technology is proud of this human genetics department because it has done a very significant contributions in several areas of research in human genetics. From its inception itself, several people, I know when I joined this university, Professor Golla Reddy, Professor Veer Raju, many others, they have done a very significant contribution. Very internationally renowned people has came to this university and interacted with these people, and still the works carried out by Professor Paddaya and his team and Professor Sudhakar, Dr. Lakshmi Garu, Dr. Kalpana Garu, and these are very, very significant. So, not only just they are doing the research for the research cause, they are also wherever, whenever opportunity comes, they want to propagate the ideas useful for the society. The studies carried out, particularly in medical genetics, they are very significant and popular. And the interaction with medical people, doctors, nurses, and hospitals with this department is very high. Really, this department is doing a very significant contribution for the development of our university, especially the College of Science and Technology. For that, I thank all the faculty members of this department for their incessant efforts to promote the culture, what it was actually initiated by the earlier people, that is continued, and the legacy is taken forward to the younger generation. And in this context, I would like to mention our beloved Vice Chancellor is very particular about this department. Whenever any occasion comes, whenever I spoke to him, there is a lot of scope for genetics department. That is, he wants to bring a new course. In India itself, he wants to start a first time BTech genetic engineering in collaboration with human genetics and computer science department and interdisciplinary. It was approved in the Senate. Most probably next year the course will be initiated and your department will be involved in technology also. in training the students. Not only that, the MOU he signed with RINL, I know how much effort he has taken for that to bring the infrastructure here. And also the laboratories, food and drug testing laboratories. And whenever I talk about the person who has to look after that is not the pharmacist alone or not a food scientist. It is the human geneticist who has to do that. That's why one full floor in the new building of 20,000 SFT is given for genetic engineering and genetic counseling. It is coming up very shortly. By next January itself, it will be inaugurated. And I am sure with the dynamic leadership of the present head of the department and other faculty members, the genetic counseling center will be started in a big way in this department for the benefit of the people not only in Visakhapatnam, in the state of Andhra Pradesh whole. And this department will be a nodal hub in genetic counseling as well as human genetics research for the entire state of Andhra Pradesh with the support that was provided by our Honorable Vice Chancellor. And I thank the organizers for giving me an opportunity to associate with this webinar. I really congratulate the young students who are participating in this webinar. They are very fortunate to hear the lectures from a very, very eminent personality like Raghunath Rao. I know him. As a doctor, even he used to involve more in academic and research. He was the president of AP Academy of Sciences. So his lecture, not only his, all the other resource persons are equally competent with him. And the young people will definitely get benefited by hearing such lectures. And I once again congratulate the organizers for doing this as a blended mode, that is offline and online put together. This will help the present students to learn more and more. Thank you. Thank you all.
One thing for your kids, sir. The first speaker is Prasad. The first speaker is Ajay Prasad Pathway. So register surrenders of this group. First speakers.
Very nice.
Sir, most important aspect of this thing, the alumni of this department are there in Harvard, Stanford, Berkeley, Caltech, Columbia, even Oxford, Cambridge. You name any university, the best university in the world, you will find one of the alumni of this department.
That's really great. Really, here I would like to mention, even though even after closing of this, we are proud Andhra University is having a great legacy of having a wonderful alumni. The alumni based support, not only having the alumni, they support the young students, whatever way they can. So we thank all those alumni of the College of Science and Technology and belongs to the Andhra University. So I thank you all.
I'm not at all surprised to hear our principal sir's message, because he has touched upon all the different aspects of human genetics, starting from the genes, polymorphisms to genetic screening, genetic counseling, and array technology also, in addition to the computational statistical analysis, because sir is from statistics department. But I am very proud and happy to reveal that he has guided several PhDs that are interdisciplinary. He has also guided PhDs from life sciences. So it's not a big surprise. He's competent enough to talk in any subject. So we are proud to have you, sir, as our principal, College of Science and Technology. I thank you very much for your best wishes for our department, and I promise you that we will strive more to keep up our opinion, your opinion, Vice Chancellor opinion regarding the Human Genetics Department. Thank you very much, sir. Now I request Dr. V. Lakshmi Kalpana to propose a formal vote of thanks for the inaugural session.
Master ఒక్కలే ఉన్నారు. Good morning to all of you. It's my privilege to propose vote of thanks for the inaugural section of this one-day webinar on recent advances in human genetics. First of all, I want to thank our esteemed Vice Chancellor, Professor P.V.G.D. Prasad Rao Garu, Andhra University, Prasad Reddy Garu, Andhra University, for his acceptance to conduct this one-day webinar in the Department of Human Genetics. I also extend my thanks to Professor K. Samatha Garu, Andhra University, and Professor V. Krishnamohan Garu, Registrar, Andhra University, for their support. I would like to thank our dynamic and beloved principal, Sir Professor Srinivas Rao Garu, Andhra University, College of Science and Technology, for gracing this occasion. Thank you very much, Professor Dr. P. Syam Prasad Garu, for accepting to deliver inaugural speech to address the audience, which will in all the audience. I extend my thanks to my teachers and retired professors of our department, Professor G. Gollareddy Garu, Professor P. Veerraj Garu, Professor T. Venkateswara Rao Garu, Professor M. Satyanarayana Garu, and Professor U. Krishna Rao Garu. I would like to thank our honorary Professor P. Peddaya Garu and the senior professors and my friends, Professor G. Sudhakar and Dr. V. Lakshmi, for their continuous effort for conducting this one-day webinar. And also I would like to thank all the other teachers of all 4 departments, Uday Kumar Garu, Mahankali, Sridevi, Rajeswari, Lakshmi, Bindya, and Sudhakar, for their continuous and constant support for conducting this one-day webinar. And also I like to thank all the project fellows, postdoctoral fellows, research scholars, and their students, and also non-teaching staff for their support. And I am very thankful and grateful to all the resource persons for their immediate acceptance to deliver a lecture regarding this webinar topic. Last but not least, I thank press and media for their support. And especially thank you very much for Lakshmi and Sudhakar Garu for organizing this one-day webinar on the occasion of my superannuation. It's I feel very happy for this webinar, and it's my privilege that thank you very much for the organizing secretary Lakshmi and her team for giving me this opportunity to deliver this vote of thanks. Thank you very much.
Thank you, madam. Now we are closing this inaugural ceremony and moving on to the academic sessions. Now the first speaker of today's webinar is Dr. Venkata Ratnala, Associate Director, Synergy Biopharma Group, USA. He is a proud alumnus of this department. Now I request Dr. T. Mahankali to introduce Venkata Ratnala to participants.
Respected Principal Sir K. Srinivas Rao Garu and HOD Dr. V. Lakshmi Madam Garu and Professor G. Sudhakar Garu, Professor G. Padaya Garu and Dr. V. Lakshmi Kalpa Madam Garu and my dear colleagues and students, good morning all of you. I am privileged to stand before you to introduce our first resource person, Dr. Venkat Ratnala, MSc, MTech, PhD, Associate Director, Synergy Biopharma Group, USA, Regulatory Affairs, CMC Scientist. He is an experienced leader with over 15 years experience in the regulated pharmaceutical industry, holding various management and technical positions in CMC Regulatory Affairs and the laboratory. And education, Dr. Ratnala completed his bachelor's in biochemistry, botany from Andhra University in 1994, Masters of Science in Human Genetics, Andhra University in 1996, and also Masters of Technology, in Biotechnology from Jadavpur University in 1998. He completed his PhD in Biochemistry and Biophysics from Leiden University, Netherlands in the year 2004. He worked as a PhD researcher in the University of Texas from 2004 to 2005 on interactions of CCR polypeptides with chemokines, as well as from Stanford University from 2005 to 8, where he investigated the structural basis for function of the GPC receptor in humans as postdoctoral fellow biochemistry, biophysics, and molecular biology. And his specialties includes expert knowledge of regulatory CMC for biopharmaceutical product development, compliance, pharmaceutical manufacturing, operation GMPs, registration and post-approval change management, CTD dossier, and health authority interactions and global regulations for licensing and maintenance of medical products. He developed a global submission plan for a subcutaneous formulation and Keytruda and pre-filled syringe. As a group leader in USB Pharma, he supported functional biology services with production of engineered cells using CRISPR-Cas9 technology, stable cell line generation, and etc. At the CPL Biologicals and Cadila Pharma Limited, Ahmedabad, he worked on experimentation and purification of recombinant multi-epitope chimeric HIV antigens 1 and 2. He authorized various regulatory documents, published several research papers and books and awards. He was awarded the Abbott Young Investigator Award and by the European Histamine Research Society, sponsored by Abbott Laboratories. And thank you for giving this the opportunity. And now I welcome Dr. Venkat Ratnala to this seminar. Thank you.
Thank you, Dr. Mahakali. It's a great honor. I feel very happy to talk to you guys, though it's like 1 a.m. in Boston, USA. Especially when I remember Human Genetics Department, Andhra University, I feel that's the basis or the fundamental place where I learned or introduced about the modern biology, biochemistry, recent genetics, molecular genetics, and different subjects back then. I really feel happy that after finishing my master's, I got opportunity to study further in MTech biotechnology and do my PhD and postdoctoral studies abroad. It's a lengthy journey, and I chose to get into industries and try to apply what I have learned. So though I was in industry since long time, most of the time I'm associated with academics and see what's going on in research. I am in constant touch with Dr. Sudhakar, and I see what he's doing at the department. And I really want to thank Professor PVGD Prasad Reddy, Vice Chancellor, Andhra University, and Professor Dr. G. Sudhakar Garu, Director, Professor V. Lakshmi, HOD, Human Genetics. I know her personally. I met her several times. And they say superannuation to Dr. V. Lakshmi Kalpana, but scientists never retire. They are called emeritus scientists from now on. So as a scientist and a professor, I don't think you generally retire, but it's just a term people use. You are associated with science so much that you always, though you will be associated with it forever, I believe. So that's science, and especially the subjects like biotechnology, human genetics, molecular biology. So these are a journey. As we humans or Homo sapiens are continuing our journey, our genes are being continued since billions of years, and most of our genome is full of viral genes and things like that. So we are continuing with several organisms and evolving as such. So today I would like to talk about, I hope I will able to present my slides. Please let me know if you can see my slides. Are you able to see my slides?
Yes, sir. We can see.
You can see that, right?
Yes.
Yeah, thank you. So I just want to talk and bring some knowledge what's going on with industries in USA and Europe, and where the trend of cell and gene therapeutics is going on. So I am from class of 94-96, MSc Human Genetics Department. As I said, and later on I pursued other studies like in biotechnology and PhD and postdoc, and then moved on to industry. But today I would like to focus more on some of my recent work, and I am associated, I am a consultant, and I work for couple of companies in the areas of biotherapeutics, cell and gene therapeutics. So I would like to just walk through a 30,000 feet view. I know that in this presentation, some of the folks are my own classmates from that batch have joined this meeting, and I understand there are a lot of juniors, my own juniors. So I do not want to go into very in-depth details, but I just want to show you the 30,000 feet view of where we currently stand in cell and gene therapeutics. So to begin with, as we all know, that we have finished the whole human genomic sequencing in the month of April 2003. And since then, there was a lot of rush, we have the enormous amount of data. Now, how do we design? How do we get better therapeutics? What can we learn from the whole genomic sequencing? So the race started on, and people, like most of the Western countries, wanted to come and design some personalized medicines, stem cell therapeutics, smart drugs, anti-aging drugs, gene therapy, design artificial organs or blood substitutes and things like that. And from that day onwards, we are still in the nascent stages of developing drugs and cell and gene therapeutics are catching up, still up to the speed, and there's still a lot of things have to be done. So I'm just going to start with very basic things. What is a gene? So we all know that we as humans are made of cells, eukaryotic cells to be precise, and we have a nucleus in every cell. And in that, we have chromosomes. And chromosomes are nothing but the DNA sequence. Later on, we will call it a gene, which is wound around a histone protein called nucleosome. And that string of DNA is called a gene. And in that gene, we have our nucleotides. So this is very fundamental thing. You all know that. So why gene therapy? So why people, earlier days, we had only small molecule therapeutics. You discover a small molecule, basically, example is a tablet or an injection, and you give it either orally or parenterally to the patient, right? And now with the wealth of information that we have obtained by researchers like all of you at Human Genetics and other labs, we have enormous wealth of human genome sequence. So there are certain genetic conditions. For example, if you see in this figure, there is a mutated nucleotide base. So in our gene or the DNA, sometimes mutations do happen, unfortunately. Sometimes repeated DNA nucleotides can happen. Like, for example, you can see a nucleotide can be like a doublet and sometimes triplet. Sometimes an additional set of nucleotides could be added to your chromosomes. Sometimes missing DNA. Sometimes even a nucleotide base is being missed. So these are the certain genetic conditions which can lead to certain diseases or examples. Like I was fortunate to work on a company called Vertex Pharmaceuticals, where we discovered a small molecule therapeutic for a disease called cystic fibrosis. So it's basically a single or few genes got mutated in this CFTR gene, basically, which causes a misfolding of this protein, CFTR protein. So that causes a lot of mucus formation in the lungs and the digestive system of the patient, of the diseased individual. Now, I should say, whoever has this unfortunate mutation in them. That's one example. There are other examples like sickle cell anemia, which has a mutated hemoglobin form, which sickle-shaped RBCs is formed, which will, involve with the flow of the blood and shortness of breath and certain indications like that. And other diseases like acute lymphoblastic leukemia, where immature WBCs and mutated DNA in cells, immune system, and mostly in childhood, there is an issue with immune system in the body. Hemophilia, again, lack of coagulation factors and ability to clot your blood, especially factor 8, like anti-hemophilic factor, and factor 9, the Christmas factor. So all these diseases are yet, there is a desperate or a cure is needed for all these patients. So the treatment options for now, still we are, some of the companies are working on small molecule therapeutics to use a small molecule. When I say it's a pharmaceutical approach where they want to fold the protein properly so that whoever the individual is suffering can breathe properly and things like that. So, but recently, there are some approved gene therapies and ongoing clinical trials which are showing to be very promising. So again, what is gene therapy? It's in this 30,000 feet cartoon, it's as simple as that. You basically take a healthy gene, either you clone or get the gene from a human cell, or you synthesize it. You just introduce into a viral vector, a safe viral vector. So you integrate your healthy gene into the virus vector with the healthy gene, and then inject into your patient. As simple as that. So what are gene therapy products? So these are generally administered as nucleic acids or viruses or genetically engineered microorganisms. So which in turn mediate effects like transcription or translation of transferred genetic material and integration into host genome as well. So how are gene therapy products used? Basically, to modify the cells directly in the patient. I'm going to show you some examples later on, so don't worry. I mean, these are like, I'm just giving you a 30,000 feet right now. And to modify the cells in vitro, and then administer to the patient. So there are a couple of techniques. There is a buzzword going on, cell and gene therapeutics. So what is cell therapy? What is gene therapy? So ultimately, when I end this talk, you might get an answer. What is it? So I'm just going to approach in a simpler way. For example, what gene therapy products are right now, which are available in the market. For example, adeno-associated virus expressing a coagulation factor, or a T cells which are modified with retroviruses to express a novel receptor. In case of CFTR, they're trying this methodology, or an oncolytic adenovirus expressing a cytokine. Luxturna, recently approved, is an adeno-associated viral-based gene therapy indicated for the treatment of patients with confirmed biallelic RPE65 mutation associated with retinal dystrophy. Patients must have a viable retinal cells as determined by treating physician. So these are really promising products already. Luxturna cures blind people. Like, you have already a product which you basically introduce the right gene into an adeno-associated viral vector, and you inject into the retinal cells of the patient, and there is a cure. So I just want to take a break and then slowly introduce how this gene therapy, or how this whole world of genetics is now evolving into different forms. Like, basically it's genetics, human genetics now evolving into different technologies like gene therapy and cell therapy and whatnot. So scientifically, if we see, there is a two-division: clinical studies on the top and scientific advances here. So from 1960 onwards, so during 1960s, definition of a hematopoietic stem cell by transplantation in mice. So then slowly, when we are in 1918, cloning of viruses and human genes and things started around those times. When we reached 1990s, engineering of our gamma retroviral vectors, and engineering, when we reached around 2000, 2003, the whole genome was sequenced, engineering of lentiviral vectors. So the development, all these vectors are very harmful if you directly inject into the human body. So they are slowly developing these vectors, safer vectors, basically viral genome into the human body, so that it is safe when we take into our body. So during 2010 and 2020, much better techniques like CRISPR-Cas, which is called clustered regular interspaced short palindromic repeats. That's the full form of CRISPR, and Cas is a CRISPR-associated protein 9. It's basically, they are a faster and cheaper, more accurate and more efficient than existing editing method DNA. So right now, around 2020s, we are in that phase where we can edit the DNA in a matter of in a day or so by adding 2 or 3 different reagents. So that's the advances we are seeing scientifically. At the same time, clinical studies as well. Around 1917, first allogenic hematopoietic stem cells kit was discovered. CD33, it's again a cell surface protein, which is used as a marker for positive collection of grafting HSC and progenitor cells and things like that. And then metabolic and storage, CNS disorders, hemoglobin disorders. And they are also looking for, as I said, you cannot inject any viral DNA into the human body, right? So they want to see it is efficacious and it is also safe. Efficacious means when you insert your gene into a viral vector and you introduce into the human body, it should be very safe. It's not that it will cause a viral disease and you lose your life or something like that. So things are evolving from both scientific and clinical aspects. So historically, if we go back and see in gene therapy in 1960s, proof of concept for virus-mediated gene transfer demonstrated by Rogers and Pruder. In 85 to 90, first gene therapy clinical trial by W. French Anderson. He is called as father of gene therapy at NIH US. In 1996, founded an American Society for Gene and Cell Therapy. And 1996, development of lentiviral vectors. This is the beginning where you are using viral vectors to introduce genes into the animal cells. 66 to 99, gene therapy vector testing in animals. In 97, first gene therapy vector tested in humans. In 99, patient death during gene therapy clinical trial. So that was the biggest full stop for gene therapy. A lot of people were having huge hope on gene therapy, but there was a death of the patient, and there were several question marks and how to proceed with it. Because if there is a case like somebody dying, it's very difficult to get approvals or a green signal from FDA to do even clinical trials. So again, but the scientists were persistent, and 2002 development of a novel adeno-associated vectors, serotypes for increased efficiency were found. And the science continued, the research continued. In 2003, Chinese approval of GenDsin, first ever gene therapy product. So Chinese were the first to discover first ever gene therapy product. And 2008 to 2009, hallmark gene therapy clinical trials of inherited blindness and central nervous system and also metabolic and storage disorders. In 2012, first European gene therapy product was approved in Amsterdam by a company called Unicure, and it's Glybera. But it costed a lot, $1 million for one treatment. And 2013, discovery and utilization of CRISPR-Cas for gene editing. In 2014, first clinical trials for genome editing and tissue regeneration. 16, first ex vivo gene therapy product approved called Strimvelis by GSK. 2017, first FDA gene therapy approval of Chimera. So I'm going to show later on little details about those products. But as I said, the major breakthrough was by Chinese scientists in 2003, GenDsin, which was the first gene therapy product, and it cost $360, that's it, treats squamous cell carcinoma when patients had mutated gene P53. In 2012, as I mentioned, Glybera, which treats a lipoprotein lipase deficiency as a genetic disorder, and it cost $1 million. And the reason for withdrawal of that product was nothing safety or any other issue, but the cost. It was tremendously expensive to treat the patient. So that's another factor. Even when scientists develop the product, cost is also a big factor, where people like common people should be able to pay for their medicine. FDA in 2017 approved Chimera, Novartis, which still costs a lot of money, $475K, which treats acute lymphoblastic leukemia by fighting cancer by using body's own T cells. So it is still expensive, but in wealthy countries like US and Europe, the insurance pays for it and people get the treatment for these gene therapy breakthrough products. So here again, I'm showing you the list of products which were recently approved by FDA, like Zolgensma for pediatric patients less than 2 years of age with spinal muscular dystrophy and biallelic mutation in the cervical motor neuron, SMN1 gene. So most of these products target disorders which are basically because of the gene mutations or the hereditary diseases. Yescarta from Kite Pharmaceuticals, they treat the relapsed or refractory large B-cell lymphoma. Most of the research is like 50% currently in US. The gene therapy or cell therapy research is focused on treating lymphoma or oncology related disorders. Luxturna, it is, these are all mid-sized companies and big companies, all are working towards gene and cell therapy products. Luxturna is again a retinal disorder where you have a confirmed biallelic RPE65 mutation. It cures blindness. And Chimera, it's a treatment for lymphoblastic leukemia. So now I want to take a break from gene therapy, and there is a buzzword, cell therapy. So let me just introduce what cell therapy or how, still there is a lot of debate how it should be called. Actually in Europe, most of these gene and cell therapy products are called ATMP or advanced therapy medicinal products. So still the nomenclature is still vague, and things will I should say the right terminology and the wording will be coming pretty soon. So cell therapy products basically are cellular therapies or phytotherapy in which cellular material is injected into the patient. For example, T cell capable of fighting cancer, while cell-mediated immunity may be injected in the course of immunotherapy. Basically, you use T cells from your own body or from autografts or allografts or xenografts, and you can try using for immunotherapy. Cellular therapy products include cellular immunotherapies, cancer vaccines, and other types of both autologous and allogenic cells for certain therapeutic indications, including hematopoietic stem cells and adult embryonic stem cells. Autologous, allogenic, and xenogenic cells that have been propagated, expanded, selected, or sometimes pharmacologically treated or otherwise altered in biological characteristics ex vivo to be administered to humans and applicable to the prevention, treatment, cure, diagnosis, or mitigation of this disease. So basically, you can take the cells from your own body or from a donor who is closely related to you, or a xenogenic cell, or completely a cell from a different species altogether, and they can be propagated, and you can treat them outside the body by pharmacological intervention, or even use the techniques like CRISPR-Cas where you can edit the DNA. I'm going to show you some of the examples what they do in cell therapy area. So examples of cellular therapy products are stem cells derived products like hematopoietic, mesenchymal, embryonic, umbilical cord blood, etc. Cancer vaccines and immunotherapies, dendritic cells, activated T lymphocytes, B lymphocytes, monocytes, pancreatic islets, chondriocytes, keratinocytes, hepatocytes, cancer cells chemically modified or unmodified. As I said, xenotransplantation products, gene therapy modified cells. For example, Chimera, I use this, this drug I use in gene therapy as well. Now you can understand. Chimera is a CD19 directed genetically modified autologous T-cell immunotherapy indicated for the treatment of patients up to 25 years of age. And Yescarta is approved by FDA. It's again a similar CD19 directed genetically modified autologous T-cell immunotherapy indicated for the treatment of adult patients with relapses or refractory large B-cell lymphoma after 2 or more lines of systemic therapy. So basically the cell therapy is something you take the cells and you do some genetic engineering outside the body and introduce into the body. So there are certain drawbacks, opportunities as well as challenges when you use cell therapy products. This is all very interesting. Oh, you can modify the cells externally and you can re-engineer them and introduce into the body. But when you try to think it as a pharmaceutical industry point of view, there are several challenges, which I will tell shortly. Some of the opportunities are the cells are dynamic. They migrate, proliferate, differentiate, and respond to the environment in vitro and in vivo. Multiple cell types and mechanisms of actions can be involved, like novel investigational products. Hundreds of INDs. IND means the clinical trial applications, initial investigational, whenever there is a clinical trial going on, people have to apply for INDs in USA. So therapeutic outcome can be curative and permanent with potential promise like repair or replace, restore or regenerate. So the challenge is, as I mentioned, for cell therapy products is no terminal sterilization. For example, when you get your injection, so all the products, when they produce in a big biotech factory, even biotherapeutics or monoclonal antibodies, they're all sterile, highly sterile products. And sometimes biologics, they don't sterilize by autoclaving or anything. They use nanopore filters and things like that. But certain small molecules, they do autoclaving and things like that. They're produced in large scale and do a terminal sterilization. But cell therapy products, they have certain shelf life. They have limited shelf life due to cell viability. So you can freeze them, but the viability may not be optimal when you rethaw them. So those are some of the challenges of cell therapy. Small lot size, limited small volume. You cannot produce huge amounts like 20,000 bioreactor liters, or you cannot produce a massive lot for a huge population or things like that. So limited availability of starting materials for process, product, and test methods and development. So these patient-to-patient variability and cellular heterogeneity is also a challenging factor. So these are some of the cell therapy opportunities and challenges. So now I want to jump back a little deeper. Now 20,000 feet view of how what the different gene therapy types are available, and how the classification is currently being done, and things like that. So basically, you can in two different gene therapy types right now, germline and somatic. For example, in this cartoon, there is a patient couple. Unfortunately, they have an issue, fertilized egg from patients with a faulty mitochondria. So they have a problem, and their egg has faulty mitochondria. So what can they do with advanced gene therapy techniques? You have a donor couple here who have a healthy mitochondria, and the donor fertilized egg with healthy maternal mitochondria. So basically, you want to introduce the genome from this patient couple to this donor's couple, donor fertilized egg. So there are techniques available. You can remove the healthy nuclear DNA from this fertilized egg and then transfer it to the donor fertilized egg, from where the nucleus of their DNA is removed. And now you have patients nuclear DNA transported into the donor egg with healthy mitochondria. And then within 6 to 8 days, an old embryo, 6 to 8 day old embryo, free of mitochondrial disease is available, which can be used again, either in donor or in a surrogate mother or in the real mother. So, or you can use it for gene and cell therapy. And there are certain instances where if there are some genetic troubles, they can be engineered at this stage as well. So that's germline, where it is, I would say, precise medical care, or very, very few patients can be treated in this way, but not a large number of people can be treated. Gene therapy using somatic message. So here, the copies of the normal human gene cloned into a bacterium, the copies of normal human gene clone, and that DNA can be inserted into the genetic material of a virus, or the DNA can be wrapped into fat-like liposomes, or even our COVID-19 recently discovered by Moderna and Pfizer use this technology, where they use mRNA and put into liposomes and directly inject into the patient. So there are two ways: either you put it into a genetic material of a virus, or these are the transport vehicles, the liposomes, and then you can introduce into the cells of individuals with the genetic disease. So either way, this is a 20,000 feet view, what you can do with the somatic gene therapy. So again, it's a clearer picture, like what I was mentioning, what you can do with your genetic material. Here is your genetic material, and then you introduce into the viral vector and directly deliver into the genetic material in vivo gene transfer, as simple as that. And either you can do that or harvest the tissue from the patient, isolate the cells of interest and do it in vitro and do your genetic manipulation. You take your DNA of interest, or if you can insert the DNA of interest into a ex vivo gene transfer, and then later on re-implement the modified cells into the human body. So now I just want to introduce another concept called gene editing. In gene therapy, gene editing is one of the processes which could be used, or sometimes you really do not need to use any gene editing. I mean, sometimes you can, it depends on disease to disease, what you're dealing with, what your area of interest is. So, but I just want to introduce the concept of gene editing, where you can, if you see this cartoon, there is one single nucleotide, the DNA with a mutated gene. Basically, you get rid of that gene, and using a tool called CRISPR, which I will introduce what it is, used to remove the unwanted mutations. And you take your gene of interest. Again, the tool is CRISPR, used to replace or repair the unwanted mutation. You get the new DNA or the nucleotide, and then introduce that the proper DNA into the gene, into the gene of the patient. So that is basically gene editing. By using certain enzymes and certain DNA, RNA, guide RNA. So basically, this is a technique where in vitro, by just adding 2 or 3 different reagents, you can, it's a more accurate and more efficient way that you can edit the gene in vitro. So this is a little busy slide, but I don't want to make it too complicated. I just want to give an example or the method how CRISPR is done in vitro. So as I said, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And Cas9 is a protein, CRISPR-associated protein 9. So basically, here if you see in this figure, DNA double helix with mutation. The pink strands are the gene or the DNA with the mutation, unwanted mutation, say we don't want that mutation is there in our cell. So now you create a guide RNA is created that matches the mutated DNA. It's basically synthetic these days. You just order a chemical guide RNA synthesizing company. You can synthetically or biologically, you can create using bacteria too. But these days they use the chemical synthetic method and get the guide RNA. And there is, and you take this enzyme, Cas9, it's a protein basically, and you add this guide RNA as well as this Cas9 protein into your Cas9 is added to the guide RNA mix. Then cells are injected with the guide RNA plus Cas9 mix. So then the real magic happens. Here, the guide RNA identifies the mutated DNA. So the guide RNA looks precisely to see where the mutated part of the DNA, and you can synthesize that depending upon, you know exactly where the mutation is. So you will synthesize your guide RNA based on what you want to target, right? And then Cas9 requires a simple PAN sequence to actually bind to the target DNA. So this Cas9 protein needs a specific sequence to actually, and so that it binds to this the region where you need to do the gene editing. So then double-stranded break in the target DNA happens because of this enzyme. It cleaves where the mutations are there, it cleaves, and there two different pieces of splicing is formed. And you take the right, the faulty gene can be replaced by the corrected DNA or a new gene altogether. So you throw in the right gene of interest, whatever you have in this break, and then you use that product to either you can use that for gene therapy or multiple other bioengineering processes, and you can use it as you can knock out some of the genes of interest as when there are certain experiments which you can do, like knockout mice, you can basically knock out the genes what you're targeting. So this is a 30,000 feet view of CRISPR-Cas technology where they are currently using in gene editing. But as I said, you do not need this technique in gene therapy. When it's needed, these technologies are being developed. So now the important challenge for gene therapy is like how do you deliver this genes into the human body? So these were really challenging in 90s and 2000s. So as I mentioned in previous slides, that there was a lot of development of using viral vectors like retroviruses, adenoviruses, adeno-associated viruses. So this is really the king right now, adeno-associated virus, where most of the companies are using this technology. I use a lot of this technology to introduce the proper genes to deliver into the human body. There are other techniques like non-viral vectors or synthetic vectors can be used. And there are other techniques like the physical methods, like by transfection by electroporation. MaxCyte is a company which does a lot of companies are using this technology of transfection by electroporation for gene therapy. And chemical technologies like lipofaction by lipofectamine. Basically, for the COVID-19 vaccine, Moderna and Pfizer and BioNTech use this technology. Basically, they have created an mRNA of the spike protein and mixed with some of these lipid-like substances, it's patented though, and then introduced into our body. So that's one of the first time they have used an mRNA into a body to develop a vaccine. So interesting days are beginning right now. So gene therapy using an adenovirus vector. So as I said, how this adenovirus vector works is like you have your gene of interest or new gene, modified DNA is incorporated into the virus. So into the viral genome, you introduce your gene of interest, or in case if you're treating a mutated gene to a patient, you introduce the right gene. And then there are certain proteins which are present on the adenovirus, like capsid proteins or penton protein. It's again is a major capsid protein which forms vertices of the capsid and interacts with certain hexons and fiber proteins when it enters into the nucleus of the human genome. So basically, you package your gene of interest into this adenovirus vector, and this virus enters through the cell membrane, and the virus is packaged in a vesicle, and increasing acidity in our eukaryotic cell causes the release of these pentons. These proteins break, and the penton toxicity breaches the vesicles, and eventually this viral genome package enters into our nucleus through our nuclear pores. So when this virus disassembles and delivers the DNA into the nucleus, so entering our nuclear membrane and delivers the gene of interest with the viral DNA, and then our transcription and translation mechanism in our nucleus ignites, and by using our internal RNA polymerase and things like that, the mRNA is produced, and later on the genetic information from the new gene is transferred to a molecule, mRNA, which serves as a blueprint for protein production. It could be an enzyme or any factor or anything which we are interested in. So now I want to walk you through into certain real world examples where I work with this company. It's called Nightstar Therapeutics. So their vision is to maintain and restore a vision. So they have a vision to treatment of choroideremia. It's a retinal disorder. It's basically, this company is headquartered in London, UK. We have an office in Boston and Massachusetts. Pretty small company, and recently acquired by Biogen USA for $900 million. So this is a clinical stage company, gene therapy company, which is focused on developing and commercializing novel one-time treatments for patients suffering from rare inherited retinal diseases that would otherwise progress to blindness. So extensive experience in gene therapy, ophthalmology, and rare diseases. And they have strong tie-ups with top research institutes in UK, like Oxford University and Cambridge University, and a lot of research has been done for this treatment. So as I said, their vision is to maintain and restore the vision in blinding inherited retinal diseases. So choroideremia disease background. Right now, it's in phase 3 clinical trials. The lead indication is phase 3 registration is going on right now. So in this rare degenerative disease, which leads to blindness with no treatment options. So it's an X-linked inherited mutation in CHM gene, which encodes a protein called Rab escort protein 1. It's shortly called REP1, which is again an orphan disease, 1 in 50,000. And there is a lot of emphasis from US FDA to treat a lot of orphan diseases. Like there are so many incentives given to the companies to work on this sort of orphan diseases. And and more these companies are trying to cure these genetic disorders. So this also falls, and they can give approvals very quickly too, because if things work, there is no therapy as such, and the FDA approves this type of therapeutics very quickly too. So this, again, going back to the science, RET1 protein plays a key role in intracellular protein trafficking and eliminating of waste products from retina. Basically, that's the protein function in the retina. Absence of this functional REP1 leads to death of RPE cells and degeneration of photoreceptors. So the disease progression is early night blindness in the early stages. In the mid stages, you lose your peripheral vision. And the late stage, you lose your central vision completely. So this is how a normal retina would look like. That's the optic disc. That's the macula. Choroideremia retina, this is one of the patients, age 57. His retina is pale due to extensive loss of retinal cells and choroid. So this is how a patient with this genetic disorder, with the loss of Rab escort protein 1 would look like. So I just want to show how the vision would how the disease progresses over time. So at the age of 17, you can see this is a normal vision when you are till 15, 16. You can see everything, but at the age of 17, early or night blindness creeps in, so your vision becomes blurry and dark. Excuse me. By the age of 23, you have mid-peripheral vision loss. So most of the peripheral vision is getting into dark. By the age of 29, mid-peripheral vision loss happens. And by 45, you can see now that only islands of vision remain. And by 55, it's already late, and the visual acuity is a complete loss. I use the term visual acuity. It's nothing but the, okay, it's a term how you use how clearly your vision is, and technically there are some numbers and things like that, but generally how your vision is, that's the visual acuity. So now how I do not want to go into very much details how this is done. Basically, I will explain again. They have taken the gene of interest, NSR-REP1 gene, and they have synthesized it and used the adeno-associated vector and cloned into that, and they are using adeno-associated vectors to introduce into the eye directly. Basically, you mix your gene of interest with adeno-associated virus, and there is a term called vitrectomy. It's an eye surgery to treat various problems with retina and vitreous. So you take a very small needle and pierce into your retina. You inject into your retina this combination of gene and adeno-associated virus. So basically, a gene is introduced just directly by a syringe into your eye. So maintenance of the visual acuity is the primary goal. So for clinical trial purposes and how FDA gets approval, so primary clinical goal is to maintain maintenance of visual acuity, meaningful for patients. So they use certain terms called regenerate is a phase 2 trial. Regulatory strategy for approval was like they should be able to see a gain when you use this treatment. They should be able to read up to gain up to 15 letters. Like you should be able to read NCKZO RHSDKDOVHR. So if you treat the patient with this gene therapy and if the patient is able to read up to the 15 letters of the 3 top lines, you get the approval. So FDA would accept it as an endpoint, and the company would get the approval for this gene therapy products. So right now, phase 3 clinical trial is going on. So I can show some of the data what has been obtained and during phase 1 and phase 2 studies. So the results are very encouraging. Over 90% of the NSR REP1 treated patients maintained visual acuity. That means 90% of the patients could be treated using this gene therapy method. So you can see here on the y-axis the percentage of eyes with greater than or equal to 5-letter loss. So using they use 2 different doses. They just called one high dose and the normal dose. So for the first 1 year of follow-up with a high dose, only 8% of the people, eyes with less than or greater than 5-letter loss. And for the first 12 months, around the sample size of 308 people, only 13% of the people had a vision loss with less than 5 letters. So this is very interesting. And in the second year, with the high dose, again, it was 8%. Only the folks, the unfortunate folks, with percentage of eyes with greater than 5-letter loss is very less, even after the 2-year follow-up. But the dose, when the dose was normal, they have not come to the right dose yet. But you see that 22% of the people have eyes with vision loss of greater than 5 letters vision loss. Basically, I just do not want to go into the in-depth clinical trials results, but what I mean to say that over 90% of the NSR RP1 treated patients maintain the visual acuity. And an interesting result they found, there are certain responders called hyper responders. Like when the patients were treated with this gene therapy, 21% of the people have gained much better response, that 15-letter gainer observed in treated patients in phase 1 and 2 trials. So like 21% of the people, when you compare it with the treated and untreated people, you can see the difference that, as I said, that the patients could read up to the first 15 letters of that chart. And if this is a great control experiment and untreated people couldn't get their vision at all. And this is again another, this is a single treatment. I would like to say that because the gene probably is getting stably integrated into the retinal cells. So the durable hyper responders, for all those responders who have seen greater than 15 letters gain established by the month 3 and visual acuity gain maintained beyond month 12, you can see that most of they have taken patient A, patient B, C, D, and D, and this is the number of months following the vector administration. And I think most of the patients, even after 18 months or even after 60 months, they gain their vision and they could read, their visual acuity is better than 15 and they could read and, their vision has stayed intact even after 60 months. So that was very promising result, data point too. So again, as I said, safety is the most important thing for gene therapy when you try to introduce this viral vectors and this gene of interest. So current data from 32 treated patients in completed investigator-sponsored trials indicate NSR REP1 was well tolerated. So no side reactions or nothing crazy happened to the patients who ever got these injections of this viral gene therapy. Safety profile profile was consistent with virectomy. As I said, it's a surgical word which is used when you do a surgery on retina or vitreous humor of the eye. So adverse events of varying severity and duration generally resolve within 1 week. So retina changes, intraocular inflammation, and visual disturbances were some side effects. There were only 2 serious adverse events, like one possibly drug-related intraocular inflammation and one procedure-related when you inject the gene DNA. Probably there was a gas bubble in the surgical tubing or something. So only 2 patients had adverse events. So with this great discoveries and great boon of gene and cell therapies, there are potential risks associated with them too. Like the therapeutic DNA, when it's injected into the cells, need to remain functional, and the host cell should be long-lived and stable. So if you are injecting DNA into the eye, you better be careful that you do not have any contaminants or any virus other than adeno-associated virus, so that you do not introduce the foreign gene, unnecessary genes into the human genome, basically human genome of the patient. Any foreign object which is inserted along with the gene might elicit an unwanted immune response. So immunology is another field where whenever you try to introduce something foreign to the body, there would be a lot of immune response to it. If that happens, rejection might happen in the patient, and repeating the therapy will be very difficult. Problems with viral vectors, especially toxicity, immunity, and inflammatory response. And there was one case when insertion of a gene at a wrong place might lead to an effect of a function of a normal gene, happened with a P53 gene, which resulted in a leukemia. So that unfortunate patient had cancer because of a wrong gene inserted at a wrong place. So as they say, with lot of powers you get, you need to be responsible too, right? So when you play with this type of therapies, the safety is the number one issue, and the efficacy of the gene and things like that have to be very carefully monitored. This is the overall slide. So now I can call gene and gene therapy and cell therapy can be basically called synonymous to one another. If you try to directly use the gene in vivo and directly inject into the patient, you can call it as a gene therapy. But if you take the cells like allogenic T cells, allogenic conjugocytes, you do some manipulation outside like CAR T cell therapy from the donor in an external in vivo condition and inject into the patient, you could call it as a cell therapy. But you can see the list is growing already. So these are all approved products in US, in vivo gene therapy drugs. Almost 24 products are already in the market. Other caveat is very expensive though. So right now, especially in US, 289 cell and gene therapy products are in development, and out of which half are for oncology treatment. Like you can see, there are in different stages of development, like phase 1 is dark blue, phase 2 is light blue, and phase 3 is in green. So oncology is one of the topmost disease areas where cell and gene therapy products are working on. And there are several other diseases like Alzheimer's, arthritis, autoimmune disorders, Crohn's disease, diabetes, genetic disorders, respiratory diseases, neuro disorders. They're trying into several disease areas by using these technologies, using advanced cell and gene therapies. In summary, I would like to say, well, folks who are studying gene genetics they're doing their bachelor's, PhDs, and research. There is a very promising future is there. It is still in infancy. various pros and cons, but still a promise for many inborn errors of metabolism. As I said, safety, purity, efficacy, and stability of these gene and cell therapy products are key for the development for cell and gene therapy therapeutics. We need to develop very tight specifications, like whatever we are using is the right gene of interest. You are not putting some additional genes or some additional viral DNA or non-suitable things to the human cells. So a lot of regulations and rules have to be developed for this cell and gene therapeutics for intended use. Very good manufacturing practices have to be done when it goes to industry scale or large scale production. And yeah, I mean, all this research ultimately aims for patient safety. That's the first priority. And the patient should be safe when you inject this type of therapeutics into the body. Well, that's what I wanted to talk about. And finally, I would like to thank the organizing committee, Professor Dr. PVG Prasad Reddy Garu, VC, Dr. Sudhakar Sir, Dr. V. Lakshmi, Dr. V. Lakshmi Kalpana Garu, Dr. D. Uday Kumar, I know him, and all the other organizing members. Thank you all. And I would like to thank my faculty, the class of 1994-96. We were taught by famous professors like Dr. Gollareddy Garu, Professor Veerraj Garu, pioneers in their knowledge, Dr. M. Satyanarayana Garu, cytogenetics, Dr. T. Venkateshwar Rao Garu, statistical genetics, great personalities, Dr. U.V. Krishna Rao Garu, he scared me with his skeletons, bones, and a lot of good genetics. Dr. G. Sudhakar was the professor who taught us the modern aspects of genetics to us at that time. And also I would like to thank my batchmates, who I'm in touch with most of them. We have a WhatsApp group, we discuss science and other things. And this class gave me lifelong friendship as well with different folks. And I would like to thank you all for your attention and having patience to listen to me. Thank you all.
Sir, this is Professor Rajendra Prasad, Principal, AV College of Pharmaceutical Science. Can I go to the question?
Thank you, Dr.
Yes, sir.
Venkataratnala. You have given
Thank you.
a very exhaustive lecture on cell and therapy. It started from what is gene, about the history of gene therapy, the evolution of gene therapy, the breakthroughs in the gene therapy, and you have touched upon the concept of gene editing using CRISPR-Cas9 technology, the different gene delivery methods that were covered in the first half of the lecture, and in the second half of the lecture we have covered with a brief account of the gene therapy that has been used for treating chorioderemia in the company you are working with. What is the gene therapy solution for that particular disease, and the underlying potential risks have been explained very well. Thank you very much, sir. And I would want to know if there are any questions that have to be posed. to the
Rajendra Prasad from principal.
yeah, Professor Rajendra Prasad. Thank you very much, sir, for joining
Yeah.
in our webinar, sir.
Well, I appreciate you staying at late night.
Thank you, sir.
This gene therapy, according to us, has not delivered to the point it is expected to deliver. Have you identified the long-term toxicities associated with gene therapy? As you said, it is one of the major limitations is unwanted immune response.
That's a very
One of the major limitations. But apart from these areas, is there any potential to treat diabetes using gene therapy? Unnecessarily, we are dumping drugs like anything to treat diabetes. It appears to be one of the promising areas. If you can come out with a successful gene product capable of treating or addressing completely the diabetes, the permanent solution. That is one
yeah,
area.
that's a very
We are looking for medical disorders, chemical imbalances, again using so many drugs, we don't know how it is acting and how it is producing toxicities. These appear to be two promising areas apart from treating cancer. What is the present status?
So, most of the companies are trying to work on the genetic disorders right now so that they want to get the ground covered. Like if they know specifically what gene to be inserted and they know if they can engineer that particular part of the gene. And the other, when you said the diabetes, so they should also look and see what type of cells they have to engineer. For example, diabetes, lack of insulin per se. And if you want to devise a genetic gene therapy for hepatocytes, we need to make sure that this gene of interest goes into it stably integrates into the hepatocyte, islets of Langerhans. So those things have to be studied, but I'm not an expert into that area of insulin or diabetes, but I am sure that people are working on those areas, how exactly they need to tackle the challenges, because insulin is something which has to produce day in, day out. A lot of protein molecules need to be generated. So they have to figure it out how exactly they can do it, and they cannot overexpress it or they cannot reduce it. So they need to have a proper balance of the right amount of gene to be expressed so that the right amount of insulin is produced. So that's a complicated thing, and I'm sure some of the companies might be working on different techniques to achieve that.
Okay, thank you, sir.
Hello, Doctor. This is
Hello. Hello.
Madam.
Sir,
I am
good afternoon, Professor Girija Shankar Garu from
Andhra University
Pharmacy.
Pharmaceutical Biotechnology Department. Hello.
Hello, sir.
Hello, Dr. Ratna. Am I audible?
Yeah, you are.
Yeah. I'm Professor Girija Shankar, speaking from Pharmaceutical Biotechnology Department, Andhra University, College of Pharmaceutical Sciences. Yeah.
Nice to meet you.
Right, nice to meet you. Actually, it is very interesting, your lecture. I just want, in case of in your gene therapy, in your phase 2 clinical trials, how do you ensure to insert the gene at the right place? Is there any validation or anything that is, because in your clinical trials?
So that's a good question. Right now, what we do is once you insert your DNA of interest into the adeno-associated vector,
Okay.
we do the gene sequencing. We need to do the gene sequencing, and that's the only specification we have. So we need to know the exact sequence of the gene of interest and the adeno-associated viral vector to make sure that we are getting the right gene of interest. So that's the requirement from the FDA.
Okay. And one more is there any integration of viral genome? Have you observed any such type of thing into that
That's another interesting question. There could be some integration of viral genome. So that's why our human genome basically consists of 70 to 80% of viral genes. So adeno-associated viral genome is okay even if it integrates into the cells of human body. So I think there will be some integration of this AAV gene as well.
Okay, that means there is safe. There is no clinical observations regarding any adverse effects.
Looks like it is safe to use adeno-associated viral genome. Yeah. Because our genome has tons of viral genome.
Yes. Right. Thank you very much. So nice.
Yes.
Sir, for inborn errors, is it possible to have the right gene insertion for inborn errors? Either to protein metabolism or in the case of the gene therapy is the ultimate solution for inborn errors.
Yeah, I'm sure, depending upon what exactly, I already showed few examples. They have gene therapy methods available for inborn errors of metabolism. So they already have, and they have the treatment available for certain disorders. That's there.
Time for the second speaker. We couldn't take more questions. So I once again thank Dr. Venkatarathna. He has set an example for all our students. Wherever the student is, they should be in touch with the department and help the department in enriching the knowledge of the fresh students who have joined. Thank you, Dr. Ratnala, for giving us lecture at this late night, as Professor Rajendra Prasad sir has mentioned. Thank you very much for your love and affection towards the department. We'll move to the second session. The second speaker is Dr. D. Raghunatha Rao Garu, who is a renowned oncologist and President, AP Academy of Sciences. Now I request my colleague, Dr. D. Uday Kumar, to introduce Dr. Raghunatha Rao Garu.
Good morning, everyone. Welcome to this session. It is a pleasure to me to introduce Dr. Raghunatha Rao Garu, MD, DM, FAPS, FRCP, FACP. Raghunatha Rao Garu got his MBBS degree from Andhra Medical College, and he is MD from Postgraduate Institute of Medical Education and Research from Punjab University. He got his MD in Oncology from Cancer Institute from University of Madras. He worked as a professor of medical oncology at Nizam Institute of Medical Sciences, Hyderabad, from February 2003 to December 2013. At present, he is the director of Homi Bhabha Cancer Hospital and Research Center, Visakhapatnam, and also chief medical oncologist in KIMS Icon Hospital, Visakhapatnam, and he is a president of AP Academy of Sciences. coming to the major achievements of Sir, he established the first medical oncology department and a comprehensive cancer care center for state in year 1989, and a subsidized cancer pharmacy and a clinical research facility in year 1992. He pioneered first phase trials in cancer, new cancer drugs, and completed over 80 clinical trials since 1993 in all phases. He is the first ACCRP certified clinical research physician in India. Started the Twin Cities Cancer Club and the Medical Oncologist Forum for the state in year 2005. He was awarded Dr. B.C. Roy Award from NCI for his outstanding contributions to the development of oncology in India. He also worked as a honorary senior research fellow of Department of Clinical Pharmacology, University of Oxford, UK, between 2005 to 2009. He authored many research publications, more than 280 papers and 12 textbook papers. His major areas of research interest includes basic, translation and clinical research in cancer, cancer education and training, cancer screening and prevention, and pharmacogenetics and pharmacogenomics. I hope this lecture is going to enrich our knowledge in personal genomics. Thank you all.
May I request you to allow me to open my...
Yes, sir. Yes, sir. Yes, sir, please.
I don't seem to be able to turn on my camera. I think you need to permit me to turn on my camera. It's all right. That's all right. Should I start my presentation?
Sir, yes, sir. Yes, sir. So you can start. Sir, please switch on your camera.
It is not switching on for some reason.
Video. Otherwise, you please switch off and switch on again. Here it is clear.
Can you see my presentation?
No, sir.
I have already shared.
No, sir.
It is showing on my screen, but I tried switching on and switching off many times the camera, but doesn't seem to work.
ఏంటండి వేరే కాన్ఫిడెన్స్ అట్లా
No, I don't see. Okay, can you see my presentation at least?
No, sir.
I'm sharing my screen.
Sir, at the bottom you are having hand symbol, sir. Side of that you are having.
Yeah, I have written that. I have very clearly said share my screen.
So you had set it entire screen or just a window?
Entire screen.
And then you highlighted the screen, sir. Then you should highlight the screen and press share button.
Can you see it now?
Yes, sir. Yes, sir.
It's coming now.
Okay. That's fine. So you can see my screen.
Yes, sir. Yes, sir. You can start now.
Yeah. Thank you very much for the kind invitation and the very generous introduction. I would like to thank my Vice Chancellor, Dr. Shyam Prasad Garu, Vice Chancellor of the Dr. NTR University of Health Sciences, Vice Chancellor of the Andhra University, fellows and associate members of the Andhra Pradesh Academy of Sciences from the university, the registrars and the deans and principals of the colleges, and the organizing committee, especially Dr. Lakshmi Garu, who invited me, research scholars, and all those of you who have tuned in to listen to this talk. You just heard an hour of Dr. Venkat Ratnala giving you an overview of gene therapy. What I propose to do is purely talk about its application in cancer diagnosis and treatment, not necessarily anything to do with gene therapy. Very often we need to understand that cancer genomics is the forerunner for a precise diagnosis, a precise understanding of how cancer can result from a normal cell. We are not sure why still, how it happens, how the body deals with the various drugs, how it deals with the environmental carcinogens which enter the body through the xenobiotic enzyme system, and how it can lead to a precision in cancer medicine. So this is the gist of my talk. So I will take you through what has happened till the early 90s in cancer care, then the transformative years till about 2010, and what is the path forward for all of us interested in the contribution of molecular biology in cancer care. 20 years ago, this is the snapshot. We were treating cancer basically simply on a histological slide, the location where the tumor is, and of course, the size of the tumor. Is it amenable to surgical removal, or is it not amenable to surgical removal? And based upon a few clinical and laboratory markers, we were adding what we call adjuvant therapy. And when we couldn't do it, we couldn't excise it, the tumor, we just gave it radiation to shrink the tumor, possibly burn it up and make it disappear, or pumped in obnoxious, I would call them extraordinarily harmful drugs into the human body, which could never differentiate anything between a normal cell and an abnormal cell or a cancerous cell. To be very frank, we now have a little over 200 treatment options for cancer medicine. And as I told you, we only had 3 basic treatment modalities: surgery, radiation, and chemotherapy. More importantly, to deliver these treatments, we had very little supportive care options, either to prevent the side effects from the treatment or to treat the side effects as and when they occur. So this is, in short, what happened about 30 to 40 years back in cancer. This is the survival graph, at least in the West. We now see that mortality is slowly coming down, and the number of survivors is slowly increasing. So from the 1990s to 2010, in the next 2 decades, we find that there is a dramatic increase in survival. This dramatic increase in survival obviously means that at least, if not everything, some of our diagnosis has happened little early on, and 2, we now have some effective treatments to offer or effective combinations of treatments to offer. But by and large, it actually means that we have understood the cancer biology in a much better fashion to devise the precise test to diagnose the cancer, to devise a treatment modality tailored to that cancer based upon its genetic alteration, and to devise ways and means of preventing toxicity to the normal cells in the human body while we effect this treatment. So it is not true that genetics is playing a role only in the treatment, but it is equally true that it plays a very important role in diagnosis and follow-up of cancer as well. So since the 1990s, where have been the progress been made? It has been made in treatment, prevention, quality of life as well as in survivorship. So we have many different therapies. I won't discuss all of them. We have extraordinarily good imaging techniques, what we now call molecular imaging. That is what is achieved by the so-called PET CT scan. So you can actually use a marker, for example, using a simple marker like a fluorodeoxyglucose, you can pick up any rapidly dividing cell in the body, not necessarily a cancer, it could be anything, but you can be very precise in picking up let us say, a neuroendocrine tumor using a tagged, a radionuclide tagged to a substrate which the neuroendocrine tumor will use and metabolize. Or you can use something which is adherent to the cell surface of the cancer cell, like the prostate-specific membrane antigen, to image a prostate cancer. It will not image any other part of the body. So the imaging is so precise that you can actually follow up your treatments as early as 2 weeks from the administration of a drug. So you can actually predict what happened to the treatment, what happened to the cancer cell by the end of 2 weeks, and you can now predict, is this patient going to do well or not going to do well? You already heard a little bit about immunotherapy. I'll touch a little bit about immunotherapy as we go along. We now know that we can use genetics for the prevention also. So I will show you several areas where we can use cancer genetics for the prevention of cancer. So especially in infection-related cancers like hepatitis B, hepatitis C, and the human papilloma virus, which are largely responsible for a host of cancers, deadly cancers in the human body. We have improved quality of life through better toxicity management and reducing the intensity of treatment. That means it is tailored to that particular patient. How many cycles to give, what drugs to give, what intensity to use, when to use, and when to withdraw is also important. And we have learned a lot about integrating palliative care, not just giving morphine or not just caring for the end of life, but many other instances through genetics of using tailored palliative care regimens. Now we know survivorship is increasing, as I've shown you in the previous graph. So we now need to know why somebody did well, the other person did not do well. Why someone else got the treatment-related toxicity, the others did not get. So one, why did they live? Why did they do well with the protocol? One, the other, why did they not do well? Three, why did they develop a toxicity and did not develop toxicity? And somebody asked about the late effects of viral. I think Dr. Rajendra Prasad Garu has asked, late effects of genomic therapy. Yes, we can now precisely observe the late effects and say who will develop, who will not. And we can use gene genetics to surveillance strategies which are very well described. You already knew about prevention through Professor Shyam Prasad's example of Angelina Jolie going in for a risk reduction surgery. So she had her breast removed, subcapsular breast removal, along with both her ovaries removed, because it is called a breast ovarian cancer family syndrome. So you can use genetics to help you tailor treatments in many ways. So if we were to ask, this is a question posed for many people in, all the attendees of the ASCO's 50th anniversary last year. And this is what the top 6 advances in modern oncology were listed as. Chemotherapy cures Hodgkin's lymphoma. That is the first step which was taken in 1965. A vaccine was approved to prevent cervical cancer in 2006. Targeted drug transforms the treatment of chronic myeloid leukemia through imatinib. Chemotherapy cures advanced testicular cancer in 1977. And in 1991, the powerful anti-nausea drugs to control major side effects of cancer treatment have come about. Finally, immunotherapy of cancer was also voted as one of the top 6 advances in medical oncology. Of these, we know that the vaccine approval, targeted therapy, and the immunotherapy of cancer are all related to gene and genetics of cancers. So let us go through the transformative 2 decades which followed the 1990s of indiscriminate use of chemotherapy to precision treatment in cancer. So this is what we want. We studied the germline variation which occurs in cancers. So we now have several genetic syndromes which can predispose you to cancer. So that was my last chapter for the Journal of Association of Physicians of India. What are the predisposition syndromes for cancers? What is called cancer proneness? So we know we have already mapped the human genome, and the best fallout from there is what can be the result of this study, which can tell you which families, which genome can lead to, which variation can lead to cancer. We also know that there can be a somatic mutation, not necessarily a germline mutation, which can lead to a cancer. And then using these two, we can personalize cancer care. Now, mind you, the germline mutation as well as the somatic mutation can occur in the same persons simultaneously. For example, if you have a familial retinoblastoma, the child is born with the germline mutation of the retinoblastoma gene in one copy, and in the other copy, which comes from the other parent, a somatic mutation occurs. So the entire retinoblastoma occurs in children less than 5 years. As opposed to that, if it is purely a somatic mutation, there is no germline mutation. So both of them occur in the lifetime of the child. The retinoblastoma occurs much later in life, typically after the first decade of life. So that is the basis for the so-called Knutson's two-hit hypothesis of retinoblastoma gene mutations causing a cancer. So this is all possible because of genetics in cancer. So cancer genomics can have many different ways of understanding, but for me, it is the advent of precision medicine era. But although we think of that as simple as that, cancer biology is by far more complex than what we have imagined. So in 2000, we know that the hallmarks of cancer were published. I'll just come back to it in my next slide. And then we know in 2005, the Cancer Genome Atlas was launched, and the genetic subtypes of glioblastoma, gastric, and other cancers were identified via the Genome Atlas, which was published, the Human Genome Atlas or the Cancer Genome Atlas, which was published. So in 2014, we finally know what are the genetic subtypes of glioblastoma, such desperate tumors as gastric cancer, which is common in North Coastal Andhra Pradesh, and several other tumors in the human body. So let me give you an example of this. This is a genomic landscape of 5,000 human cancers. All the way on the right side, you can see the upper GI tract, skin cancer, sarcomas, prostate, pleural tumors, pancreaticobiliary, ovarian carcinomas, all the way to acute leukemias. So, and the genes, so-called genes studied, which are available on the x-axis for you on the side. So you can see the genes which are available for you. So it looks like some genes are commonly mutated in many cancers. Mark my words, the same gene is mutated in many cancers. And some genes are exclusive to certain cancers. And some cancers are extraordinarily bland. So it is important to understand that we have still not had a chance to know whether this is an innocent bystander mutation or it is a mutation which we call a driver mutation. So this is an example of now what I have given. So we now know that we have a spectrum of oncogenic driver mutations in non-small cell lung cancer. So there is a huge spectrum. In 55.7%, we still haven't detected a mutation, but in the others we have KRAS mutation, EGFR mutation, and so on and so forth. And we have a drug for each one of these mutations, either in the clinical trial or in the clinic. So all the way to the clinic. If we study only the EGFR mutation, so when you start a drug directed at an EGFR mutation, you suddenly find that the EGFR mutation itself is heterogeneous. It is not as if the EGFR mutation is the same one in every one of them. Although by and large we have the exon 19 deletion, then we have the T790M mutation coming up, which can be the driver mutation, which is induced by your treatment. So you give a drug against the exon 19 mutation, you develop a T790M mutation for which it becomes resistant. So it changes the small configuration for the key to fit into the lock at the locking site or the docking site. And then we have several other mutations. So our research is now driving us to find drugs for the T790M mutation. We already have 2 in the clinic, already available and manufactured within India. Well, as opposed to that, you take a skin cancer called melanoma, which is popularly seen in white people who leave their temperate climate and move to the tropical or the subtropical or the equatorial area. God gave us black color not because he hated us, but to protect us from the ultraviolet rays of the sun. Whereas he gave them a light color so that they can at least absorb some ultraviolet light to prepare vitamin D. So that is the basis for the coloration as we go towards the equator. Not any race or anything. It is a natural selection process. So it is to protect you from the ill effects of harmful effects of ultraviolet radiation. So here are the driver mutations which we find in melanoma. So you can find still in 35% we don't find a mutation, but by and large we have either the BRAF mutation or the NRAS mutation. And in the BRAF mutation, just like what occurs in the EGFR mutation in non-small cell lung cancer, we can find several subtypes of mutations which can occur. V600E is the commonest mutation, followed by other less common mutation. So we will start targeting drugs and using in the clinic for the V600E mutation also. So this is what I meant by the hallmarks of cancer. Cancer can actually, these are the therapeutic implications which we need. I'm not worried about at cell biology level. We have means of tackling any one of these pathways or all of these pathways simultaneously or in sequential pattern, starting from the cell cycle, then through immune activation, then through telomerase inhibition, then to anti-inflammatory drugs like aspirin, then through cell cycle receptor signaling, or through DNA repair mechanisms like the PARP inhibitors, or promoting apoptosis when the BCL2 gene is overexpressed like in many leukemias, or improving upon and driving the cell through aerobic glycolysis instead of anaerobic glycolysis, or through cell separate receptor blockade like EGFR inhibition. So that is what brings us to the phase of the targeted therapy in cancer. So all this is possible only because of the effects of study of genetics of cancer. So that leads to precision medicine. So we now know that we can now classify, instead of using a histological classification, we can actually classify cancer through the molecular abnormality and the site of origin. Sometimes you find the same molecular abnormality occurring in the same cancer wherever it occurs. For example, a small cell carcinoma of the lung is the same as the small cell carcinoma of the cervix, is the same as the small cell carcinoma of the skin, is the same as the small cell carcinoma of the tongue. It doesn't make any difference. So the molecular abnormality seems to predict what is the histological pattern with which it presents and what is the clinical course of this disease and what is the treatment which is required for this disease. So it is ultimately the phenotypic expression is determined by the molecular or the genetic abnormality which is present in the cancer cell. So we now have exceptional successes when treatment is matched to a driver mutation. Here I have given an example of all those which have been based upon using a mutation analysis. For example, we got the first targeted drug, a monoclonal antibody, rituximab, in 1997, largely manufactured in India. Now 10 companies manufacture it. Trastuzumab, 1998, the next year, for breast cancer. And now we now manufacture it by the dime a dozen in India. And of course, imatinib, which was introduced in the year 2001 by Professor Drucker for a molecular abnormality in chronic myeloid leukemia, as well as in gastrointestinal stromal tumors. Now, that is the paradigm shift which has occurred, which actually led to study of how to switch off the gene expression when there is a reciprocal gene rearrangement or a overexpression of any genetic information which is being read. So this is a prototype of the drug which we can now use to treat many diseases. We have come a long way in imatinib. This year we are celebrating 2 decades of imatinib use in humans. I had the chance of meeting the first woman and the first man who have ever used imatinib in Drucker's own clinic in Portland, in Oregon State in the US. And it is a pleasure to see that the first woman is now a grandmother, and the first man also is married. He has his own children, grown-up children, who are studying MBBS. So it's a matter of pride that we have come a long way for a leukemia which is untreatable in the past to a time when we can now learn how to stop treatment. How did we learn how to stop treatment? Simply because of the precision with which we can measure the gene product, that is BCR-ABL fusion oncogene, can be measured using molecular diagnostics to a precision of 1 in 1 million cells. So that means when we reach the undetectable level, we can very safely stop giving the drug any further to the patient. So we converted the disease to something called a chronic phase, to a phase where the disease is no longer noticeable clinically, to a phase where it cannot be picked up by the most precise molecular technique available to date. And now we have a little over 100 or maybe a little over 200 FDA-approved targeted therapies for cancers, in various cancers available. So this is a timeline which shows from 1997. On top, I have shown you what are the genetic abnormalities which have actually helped pave the way. And on the bottom are the list of drugs which can be used against the genetic abnormality which was discovered there. So you discovered the EGFR mutation in lung adenocarcinoma. You discovered a drug for that. That is the discovery of the gene alteration led to the discovery of the drug and to its clinical use. Sometimes it so happens that when you are screening a drug, you suddenly found a patient who was hopeless, who got helped by the drug. In fact, ALK discovery occurred like that. Here is ALK discovery. So we discovered that in a group of patients, only one of them actually responded to treatment, whereas the other 13 in that cohort actually died. And the response of this single person was so dramatic that it did not work in the other 13 of them, but one of them worked. They went back and studied his the gene abnormality, and to their surprise, they found that it was an ALK-positive lung cancer. So you discovered a drug for the ALK-positive lung cancer, and believe me, it is very gratifying to see a patient who is gasping for breath come into your clinic. You start this oral medication, and in a week's time, the patient has no evidence that they have cancer at all. The lungs are just cleared of all the cancer which is available in the body. And now we have, as rightly said, we now have immunotherapy, a host of immunotherapies available to us, and we now have any number of immunotherapies available to treat n number of diseases which are not disease specific. They can be used in many diseases, many cancers simultaneously. It is not very true that you actually develop a drug and there is no innocent bystander effect. Like the same thing which happened a little bit with chemotherapy, to a large extent with chemotherapy, can happen both with monoclonal antibodies, with the NIPS, that is the small molecules, as well as the so-called immunotherapy or the IO blockers, so immune checkpoint blockers. So these are photographs of the front and back of a patient of melanoma. You can see that after 15 weeks of treatment, there is no disease. That's just less than, they're all melanoma all over the body. You have thousands of melanoma, metastasis over the skin, and the entire thing disappeared. This coloring actually is a lightening of skin which occurs with many drugs which target the EGFR molecule. So vemurafenib was used, and the color actually changes. The color change precedes the disappearance of the disease, and that is very interesting. But well, the effect did not last beyond 2 years, and once again the disease is back wherever it was available in the beginning. So what happens is, as you use the drug, the cancer cell is a little more intelligent than the average clinician, and so it learns to bypass the pathway which was blocked, and it uses a redundant pathway or redundant pathways to make itself grow. So it knows how to manipulate the biochemical pathways and genetics and epigenetics in the cell better than any one of us. So that is exactly what it has done, and the entire tumor is back. But that means you need to study this patient once again at this level. So take a bit of this and then reanalyze the patient's tumor to know what is wrong and what is the targetable lesion at this stage. Next, we are very familiar with this ethnic diversity in drug effect. Like you know what pharmacokinetics is and pharmacodynamics is. One is what the drug does to the body, and the other is what the body does to the drug. So both are equally important to study. It is not as if the same patient works, for the same disease across the racial populations in this world, every drug works. In fact, we were the first one to pick up that erlotinib through the IRISA trial. They lost their right to market the drug when in an expanded cohort, there were many countries, 134 countries which participated in the trial. And when the data was submitted to the US FDA, it said we are withdrawing the permission to market this drug for non-small cell lung cancer. We said, hey, wait a minute. All those in East Asia and South Asia actually did very well. So we did a cohort analysis of all the patients from Japan through Taiwan, through China, through Hong Kong, and through Singapore to India to Saudi Arabia. We analyzed these patients, and to our surprise, we found that the EGFR mutation EGFR expression on our patients was very high. And so we formulated a rule that if you're an Asian, if you're a woman, if you're a never smoker, and you have an adenocarcinoma, 99.9% you are likely to respond to this drug called erlotinib. And we knew that this drug works much better in the Asian population as opposed to patients in the Western or the Caucasian descent. So there are environmental differences, local practice differences, drug action in differences. So genetic differences are even more important. So there's something called pharmacoethnicity or ethnic diversity in drug response or toxicity. So precision medicine is not just giving the drug, but also in studying what happens to the drug within the body and how does the drug react, the human being react to the body and the drug work in the human body of different races. So we can do it at a cellular level, like biospecimens and molecular pathway analysis. We can do in silico biomarker validation and multiplex assays. So you build a platform to be used as a companion diagnostic tool, and you can develop instrumentation and informatics to analyze all the data which comes from precisely the previous methodology which you have used, that is the multiplex assay, because the amount of data which comes is phenomenal. So that is where you get the science of bioinformatics, and then we can actually use it for clinical decision support and patient monitoring as well. Systems biology is a system which actually integrates all this information. So you have a tumor, you have a blood sample, so you analyze the phenotype of the patient, that is the somatic expression. You analyze the germline of the patient through a simple blood sample. You also form a mouse model or an in vitro model and analyze through a xenograft, expose it to in vitro study to what's the best drug to choose for this patient, either in the petri dish or in an animal model, and then you classify it histologically using genomics, transcriptomics, or metabolomics, what are proteomics as you call it, or omics study you do. So apart from using an image-based classification like mammography, radiography, and PET CT scan, and integrate all of them into an integrated genotypic, phenotypic stratification of a simple tumor like breast cancer towards, and you can predict what's the clinical outcome going to be, what's the response signature to watch for, what is the resistant signature which might emerge, what are the adverse effects of this signature, and then you use all of them to tailor your treatment for that particular patient to have the best quality of life with the highest survival rates. So that is the systems biology approach to a cancer therapy. Well, there are many chemotherapy advances as well. We know that from 1998 through 2015, so we have moved quite a long way for using chemotherapy appropriately for both by increasing and decreasing the treatment aggressiveness simply to maximize the quality of life, maximize the benefit. There are two precision expectations we have listed here: improved quality of life with the highest survival rates. Yes, we now have survival rates where, as I showed you, they can go into becoming parents and grandparents, and that is what we want. We have the mortality of all pediatric cancers. So we now have a huge generation of pediatric cancer survivors who are going to be available for us in the world. As I said, it is not as if only the therapeutics has taken the leap, but imaging has also taken a leap. So we can diagnose it at the earliest stages, and we have a better picture of the disease spread, and we can help patients guide through an optimal treatment. So this is what we call a rapid response assessment. Baseline GIST resistant to imatinib. After 1 week, we shifted the patient onto sunitinib therapy. So the tumor has disappeared all the way. This of course is the heart, and the tumor has disappeared from all over the liver. And after 2 months of imatinib therapy, nothing else remains in the body at all. So this is what we call a rapid response assessment. And you have heard a little bit of immunotherapy. So these are recalcitrant cancers, and this is a game-changing discovery that the T cell in the body is prevented from recognizing a cancer cell simply by the expression of a surface marker which blocks the receptor on the T cell from recognizing it. So we know that is what we call the programmed death ligand. So we now use many drugs like ipilimumab for melanoma. Then we have pembrolizumab, nivolumab approved for melanoma. Now we have many small molecules as well as monoclonal antibodies for many cancers I have listed here: head and neck cancer, melanoma, lung cancer, kidney cancer, gastric cancer, bladder cancer, uterine cervical cancer, and several lymphomas as well. So all of them are now available as the game changers. They were now originally used in recalcitrant disease. They have moved to frontline in several diseases. That means they are the drugs of choice to be used in the disease, no longer chemotherapy or anything else. So an example of this was given by my predecessor of what CAR T cell therapy is about and how to develop a customized vaccine using gene therapy. So what you do is you reprogram the T cells to recognize in the body while exposing them to a molecular soup or a drug soup or genetic soup in a petri dish, and reconditioning them asking them to relearn as if going back to the kindergarten to recognize the molecule presented to it by the dendritic cell, which is co-grown in the petri dish, and then reinfuse them back after expanding the pool of T lymphocytes back into the body, and you can treat really recalcitrant leukemias through this methodology. So we also know that infections can cause cancers. So this is the infections which are caused by cancers. If you move to sub-Saharan Africa and beyond, 33% or 1/3 of the cancers are caused by infectious diseases. So one of the most important infection-related cancers is human papilloma virus, which can cause cervical cancer, anal cancer, vulvar cancer, vaginal cancer, penile cancer, as well as oropharyngeal cancer. So taken together, it is as big as the tobacco epidemic. So one single way of doing is screening them for the human papilloma virus DNA. So you can now take a small cervical smear or a oral smear, as the case may be, and then run it through a diagnostic text on your office table. In just about 45 minutes, you will get whether the patient is HPDNA positive or negative. That's the best screening modality available for a woman in India. And of course, you have a vaccine which is introduced in India. So if you can actually use the vaccine, you can eliminate cervical cancer from India by the year, at least in the next 30 years, you will be able to eliminate cervical cancer from the face of the earth. So it will become the first cancer which can be eliminated, just like how we eliminated polio and smallpox through vaccination. We can eliminate HPV vaccine. The vaccine is very stable, and the immunity built by the vaccine is even more stable. It's not like the COVID vaccine, so where there's going to be a mutation. So we can produce stable levels of immunity in premarital girls and boys who can be vaccinated. Another cancer which can be prevented is, of course, hepatitis, infection hepatitis B. We now have a vaccine, and we know that global vaccination, which is now introduced in India, also in the pediatric age group, can actually give you a good coverage and prevent hepatocellular carcinoma or the preceding chronic active hepatitis, chronic persistent hepatitis, and cirrhosis of the liver. All the three conditions can be prevented by the hepatitis B vaccine. As I said, there are many advances in supportive and palliative care, which are also very important. What we learned is you need to integrate palliative care early on the day one of treatment for the patient. And we now know that the game changer, as I told you, has been this drug called filgrastim, once again, which is developed by recombinant DNA technology. It is the granulocyte colony stimulating factor, which helps you prevent the fall in counts and inevitable death if the patient's count does not recover in the next 24 hours. So the counts drop to 0 with chemotherapy. So using this drug, we are now able to do stem cell transplants, mobilizing stem cells either autologously or allogenically, as the case may be. So we have 32.6 million cancer survivors worldwide. So we have plenty of survivors who are available, but who now pose a different challenge altogether. They have some ill effects from the late effects which are related to our treatment. They have some effects which are on the psyche. They have effects related to their sexual health. And of course, they have disparities vis-a-vis their employability and their opportunities for marital life. So all of them are equally expanding fields of survivorship, and we should be able to help them through survivorship as well. So the path forward I'll quickly take you through is more basic research. So, and use the smarter clinical trial. I'll come in a second to that. Increase the population diversity in research, and more efficient biomarker validation is what is needed. 2, enormous possibilities, advanced health information technology. So you need to leverage what you call marry sciences together from the from the information technology into biology, and then you will learn. You need to learn from every patient, not just the patient who succeeded, but even from the patient who failed, not from the patient who was in the clinical trial, but from the patient who was not on the clinical trial. And you need to support a decision making, and you need to help them improve quality assessment. So these are all the areas of what we call aggregating information from the scientist, from the clinician, from the physician, the researcher, the patient, molecular disease models. There is plenty of data always coming through again and again and again through the tests, as well as the outcomes of the tests you have done or the treatments you have given. So you need to evolve a rapid learning system to integrate all of them. So this is very important. That means the future belongs to that person who can actually manage data and large volumes of data at that. There's a huge variation in the developed countries as regards knowledge and means to access to high quality care. That means we need to provide something. Many of us think that cost is a problem only in India. Cost is a major problem in the United States. In fact, it is now swallowing all the budget of most of those countries which offer free health. For example, the whole of Europe, Australia, Japan, New Zealand, and England, which have the National Health Service, cost is the major issue for them. In India, we are tackling cost through indigenization of the processes of everything, from the diagnosis to the manufacturing of the drug. Our industry, pharmaceutical industry, I must say, just like how we were proud that anywhere, any nook and corner of the best universities in the world you go, you will find Andhra University graduates of genetics, Department of Genetics. You will also find Andhra University graduates of pharmaceutical sciences. So that is our richness. We have contributed that much to the world. So it is well within our power to reduce the cost in our country. Fourth, focus on the needs of the whole patient, not just one point, not just diagnosis, not just basic research, not just treatment. We need to, the entire spectrum is important. From prevention to palliative care through treatment and survivorship is equally important. So keep pressing forward. Imagine where we'll be in another 20 years. So there are 3 revolutions which have happened. Cancer panomics, as I said, big data. The second is the big data, how to manage big data. And wedded to that, both of them is value or quality. You need to have both quality and value added to these 3 revolutions. To this, you add the 4th revolution, that is what we call patient engagement. So you need to engage the patient at all levels, at all times, right from the time he is healthy, through the time he is not healthy or ill, and through the time when they become completely all right. So all the 4 areas are as important to us as it is to the governments. So it is up to the university to design all these data together, marry them together, and enrich them and come out with solutions for such pressing needs. So I end this with the world famous African proverb: to go fast, go alone. To go far, you need to go together. So, ladies and gentlemen, progress always and shall always depend upon collaboration, and collaboration is the key to success. Thank you very much for your patient hearing.
For your excellent thought-provoking lecture, but the basic problem is Bert Wagelstein, the founder, discoverer of P53 gene, analyzed all the cancer-related research published in USA. He did a meta-analysis. He did another meta-analysis by taking all the published literature that is available on cancer throughout the length and breadth of the world, and his conclusions are 10 to 15% of the cancers are only have some genetic component. The rest of the 85% of the cancers are we made ourselves. One most important factor which is causing these problems are adopting Western food practices. Our practice, our Sanatana Dharma practice of food is, if we practice that, 85% of the cancers of the whole world can be stopped in their tracks. Then, see, our Sanatana Dharma said there should be enough gap between one meal and the other. In every individual's body, you'll find some rogue cells. And these rogue cells will become eventually cancer cells because of our food habits. We are feeding these rogue cells selectively to make them cancerous. And intermittent fasting. మనకి ఏకాదశ ఇవన్నీ పెట్టారన్నమాట. ఇవన్నీ ఎందుకు పెట్టారంటే even if there are some cells, rogue cells turning into cancer cells, they'll die. And 85% of the cancers can be stopped unless if we actually, because one is diabetes leading, this diabetes itself is leading to heart diseases and also to cancers. So diabetes is the thing. In diabetes again is because of indiscriminate use of eating habits and antibiotics. We have to concentrate on these because pharmaceutical and medicine and the food industry put together in the West is a multi-trillion-dollar business, and they are promoting this business by giving all types of drugs and all that thing. And we have to take enough care not to succumb our continent to cancers. Thank you very much, sir.
Thank you for this observation, sir. We actually participated in 2 very large trials. In fact, they are the largest trials ever done in public health and cancer. So one was in breast cancer. Indians in India, along with Indians, first generation Indians who migrated to Britain, second and third generation Indians. We did this with the University of Oxford. The second was a study which we did it with many Commonwealth countries. Once again, the largest contributor was India, in our dietary habits and colon cancer incidence. So what we actually analyze is what is called the total salicylate in the food level. So you can actually measure the salicylate using simply a stool sample. So I take a matchstick head amount of stool sample, and I chromatographically I can analyze what is the amount of salicylate in that food sample. So we did this again for Indians who are vegetarians, Indians who were exclusively non-vegetarians, Indians who are eating part of their meal as vegetarian and non-vegetarian in a given week, and compared it with the Indians who migrated to the United Kingdom and then adopted a Western diet. So as you move away from a vegetarian diet into an exclusive non-vegetarian diet in India and further on to a non-vegetarian diet exclusively in the Western world, or a preserved diet of the Western world as we call it, the salicylate level dramatically drops to actual 0. So the correlation between the incidence of colon cancer in India and the dietary aspirin or salicylate level is phenomenally strong. So it goes without doubt, saying without doubt that what the observations you have made about diet and lifestyle changes of aping the West are typically pro-carcinogenic. You're right, sir.
You told us that 36.5 million cancer people have survived according to your data. But according to my opinion, that the survival, even though the patients may be survived, the lifespan of the cancer patients have been reduced, I think so. Can you tell me how many years they have survived?
So there is a difference in the way we look at the survival. So these are pediatric cancer patients who survive. So we are not counting the adult cancer patients who have survived after their treatment. If I treat a 93-year-old man and he survives another 3-4 years, it won't make any difference. So for us, survival means that he should have been treated before he left his teenage and then survived into adulthood for at least 5 years or more. So, for example, it is variable. In an acute myeloid leukemia, the survival of 2 years is taken as a cure. It is unlikely that they will ever get back the disease. As opposed to that, in a breast cancer and in a colon cancer, an average survival for a cure should be 20 years. That means the patient should be alive for the next 20 years to be called cured. That is the importance of understanding what survival means. So we now have data in all those patients. As you very rightly pointed out, they may not actually die from cancer. I showed in one of my slides that cancer patients after their treatments are highly prone for various physical disorders, as well as endocrine disorders, as well as psychosocial disorders. These are important to understand. So all of them together is called health. It's not as if you are physically fine, it is health. You must be physically, mentally, psychologically, economically, socially, and professionally be healthy. Then only it is total health. So that is very coming to be as an observation in the survivors. Your observation is very right. They suffer from greater hypertension, greater diabetes, greater heart disease, and premature death from any cause. So it is important to study why it happens so in cancer survivors. What is the genetic basis of that?
Oh, thank you very much.
I see Professor Annie Hassan here. Very glad, madam, that you could join. I'm very grateful for your patient listening.
Whenever you are talking, I'm always there. I'm very impressed with your work. And really interesting combining the importance of genetics.
Thank you very much, ma'am. If there are no further questions, shall we end this session, madam? And we can reassemble after lunch for the last.
I think Dr. There is something regarding this.
Good afternoon, sir. This is Sunil from Loknaik Hospital. I'm having a question, sir. So it's very wonderful to talk from you. Sir, how about the gut microbiome analysis for cancer?
Yes, we now know that the gut microbiome can be related to several far-flung diseases which are in no way related to the gut. We know that gut microbiome is different in many cancer patients. It is altered in many cancer patients, probably a decade or two before the cancer is actually takes its onset. So it is clinically or test-wise detectable. Not just cancer, it can be as far removed as bronchial asthma, rheumatoid arthritis, SLE, heart disease, or even psychiatric disorders. We know that all these seem to have something to do with the many precursor amines which are released by the gut microbiota. What we also know is that the neutrophil, just like in the earthworm, and actually leaves the gut mucous membrane and wanders around in the microbiota, in the feces which we have, and then returns back gathering some information from there. So we know that in the large bowel we have Peyer's patches, which are actually something like the tonsils in your throat. They are information processing areas or antigen processing areas. So they actually process the antigen from available from the environment. In the upper aerodigestive tract, just like in the lower aerodigestive tract, the process. For example, we now know that the appendix is equally important. So removing the appendix or tonsils can possibly make you more prone for many disorders because you have removed the antigen processing factories in the body. So the gut microbiome actually influences this T cell function and B cell function in our body. So we know that in the past we did not believe in immune surveillance. We now strongly believe in stimulating the body's own immune system to hunt out and get rid of its own cancer cells on its own. We are actually tailoring treatments to do that. So it makes sense that the gut microbiome may actually predict, study of the gut microbiome may actually predict several cancers far removed from the gut itself. That means they need not be related to the gullet or the stomach or the pancreas or the intestines. They may not be related to them. They may be related to a cancer occurring somewhere in the ovary or the uterus or the liver. So that is the essence of what we know, or it may be related to a lymphoma or a leukemia. So we know that the gut microbiome definitely influences the signal processing by the antigen presenting cell and the response of the T and B lymphocytes to the gut microbiome dictates many diseases apart from cancer.
Thank you, sir. Thank you very much, sir. And one more thing, sir. How about breath analysis for identification of cancers? Breath analysis. Any metabolites can be identified.
That is a technological leap which you need to know. If you have a nano analysis, that is, if you can analyze in the breath, many molecules, just like how they come out in urine and stool, or can be detected in blood, can be detected also in saliva. Also, they can be detected in the breath. So it is possible to analyze, use a breath analyzer, but it is still a research tool only. So it is not yet in clinical application whatsoever.
Thank you very much, sir, for your wonderful insights and explanation. Very much.
Hello, sir. Good afternoon, sir. This is Radhika from Bandra University Biochemistry. Sir, regarding nano delivery of any of these drugs to the target site, how far it will be successful in the present scenario without affecting the healthy cells or healthy tissues, the neighboring areas of the cancer, especially
So,
in the.
We need to understand that there are two aspects for drug delivery. One is through the system, that means through the blood vessel. It needs to be carried.
Yes.
That means the vehicle to carry it is equally important. The second part is its transport into the cell and preventing the drug from being kicked out of the cell. Majority of the time, the cancer cell learns how to just put a big pump and pump out all the water. For example, from under the Chaur-Madum bridge, how they pumped out water. So the cancer cell also becomes very, very efficient. The PPP pump is just exactly that. NDR gene is actually a pump which drives out many materials out of the cell. So what we need to design is one, the efficient delivery to the site. So that you can use by tagging on to many different vehicles. That means you need to find out what is the best target available to attach to a particular given cell without any off-site delivery of the drug. Okay, so that is important. And then you can deliver the drug in a nano formulation so that it is going into the cell and then it is retained in the cell to act where it is needed. All of them are important. So we are now studying now, for example, exosomes. Many people are now studying exosomes to diagnose many diseases and using them for delivery treatments. Exosome delivery is now coming into even possibly into COVID management also. So soon we will have exosome delivery models in management of the so-called cytokine storm in COVID.
Yeah, sir. Nanoparticle delivery systems using these plant extracts and all. So how could we able to see that the neighboring tissues are not at all extending this infection by targeting that only the cancer cells, especially in the case of melanoma situation?
So you need to be targeting a particular epitope on the cell which is not shared by its neighbor. That is important to understand. So that sort of exclusivity is a little difficult to come by in most cell systems.
Okay. If you conjugate antibodies to that and you can deliver that.
Yeah, you are very right. Absolutely right. You can conjugate.
So thereby it can reach to the target site and
yeah,
there
you're
may
right.
be the possible.
Absolutely right, madam. Yes.
Yeah. This. So thank you, sir. Thank you for the insightful talk, and you are all learn from you, and we are all learning the similar way. The different aspects of these delivery systems also, but we need to have a good collaboration, as you said just now. So we are waiting for the suitable collaborations to complete the work in a progressive way. So thank you, sir. Thank you very much.
Thank you very much, madam.
Sir, Dr. Raghunatha Rao Garu, thank you very much for accepting our invitation and for delivering an invaluable lecture on cancer genomics and precision medicine.
Thank you very much for the invitation, madam. Thank you.
Sir, you have very beautifully explained how there is a transformation in cancer treatment since the last 2 decades, where indiscriminate treatment was used for treating cancer, and how currently we are using precision medicine and the significance of genetics behind that. Only because of that it was possible. Like you have told that the diagnosis and treatment of cancer are now precision based treatments, which was possible because of genetics. And you have also explained that in future, these cancers which are based on these infections, they are going to disappear just like polio. You have emphasized the integrated approach that is needed for the treatment of cancer. Thank you very much, sir.
Thank you very much for the invitation, madam.
Right. I will close this session and we'll start once again at 2:30, the third academic session.
అంటే మీరు మంచి మూడు సబ్జెక్టులు చెప్తా ఉంటారు కదా అది మీది చెప్పేసాడు ఫస్ట్ రెండు ఫోటోలు చేసి ఆదివారం సండే ఆర్టికల్లో డబ్బులు వచ్చేస్తాయి చూసి ఎవరు ఎన్నో మంది రాజస్థాన్ అలాంటి మధ్య వేయాలి అన్ని మీరు ఎట్లు చేసుకుంటారో పదవుల్లో లేకుండా జగన్ కూడా ఆలోచిస్తారు రెడ్లు ఆపేయండి లంగాట్లాగా అవుతుంది కదా వేయాలి సార్ వేస్తాను మళ్ళీ కూడా ఎందుకు అదే అధికారికి డైరెక్ట్ గా అడుగుంటాను నాకు ఉంటాను వచ్చింది సగం ఉంటే చెప్పాలి దూరంగా 30 అది కాదు 30,000 40,000 ఇస్తారు ఎందుకు మాకు కావాలా ఎంతమంది అక్కడ కూర్చొని ఇది పోతున్నాను అప్లై చేసుకున్నాను అన్నీ పెట్టాను వద్దులేండి సార్ కటం సార్లు తీసుకెళ్ళి అధికారి అంటే ఎలా చేయాలా ఏంటి హజ్ యాత్ర అన్ని చెప్తూ ఉంటారు ఏం చెప్పలేదు కదా సంబంధం లేదు మామూలుగా చూసి మీరు పంపించారని చెప్పండి సార్ వస్తున్నారు వస్తున్నారు మూడు సబ్జెక్టు
కలబడదాం ఒకసారి కలబడదాం అంట అలా ఇంకా మనం పెట్టిన తర్వాత ఇంకెవరు పెట్టారు, మనమే తెలుసుకోవాలి. మధ్యాహ్నం లోపు వచ్చినాడు సరే రైట్. ప్రశ్న చూసారు సెలవు వచ్చేస్తాం. ఇప్పుడే చేస్తాం. భోజనం
Sir. Sir.
ఎవరు మేడం? ఎవరు మేడం? మితాశ్రీ. లాగిన్ అయ్యారు. మితాశ్రీ మిత్ర ఉన్నారు. మేడం మైక్.
Hello, I am here.
This session is Professor Mitashri Mitra, who is a retired professor of technology from Patna University. I invite Professor Devi to introduce the session. Good afternoon.
Voice is coming good.
Welcome you all to the afternoon session of today's webinar, Recent Advances in Human Genetics. Most respected patron of our webinar, His Excellency Professor P.G. Prasad, Vice Chancellor of Andhra University. And today's honorable speaker, His Excellency Professor Dr. P. Shyamprasad, Vice Chancellor of Indian University of Health Sciences. And respected Rector, Professor K. Samta, Eminent Principal of Science and Technology, Professor K. Srinivas Rao, Revered Guest, Professor V. Krishnamohan, and today's international webinar secretary, organization secretary, our head of the department, Professor V. Lakshmi Madam, and the director, Professor Sudhakar, and associate director, Alpha and honorary Professor Sadhya sir, and our colleagues of advanced one. In this afternoon session, I warmly welcome Professor Mitashri Mitra Madam. She worked and retired as professor and head in the School of Studies in Anthropology at Pandit Shankar Shukla University, Rajkot, Chhattisgarh, India. Madam was a gold medalist in MSc Anthropology in 1977. Please give a clap. She was awarded JRF and SRF of UGC during the period 1978 to 1980. She has 40 years of experience in teaching subjects like anthropology and forensic science. She has extensive experience in research designing, project writing, executing population surveys in remote tribal areas and underprivileged groups in Chhattisgarh, particularly in reproductive health and women empowerment and child malnutrition. She also established state-of-the-art human genomics lab and forensic lab in the School of Studies in Anthropology, Department Pandit Shankar Shukla University, Rajkot. She published 134 research papers and authored 6 books. It's a nice. Her areas of research interest include tribal health, nutritional,
socio-cultural, anthropometric, and genetic dimensions of health, tribal studies in biocultural perspective, and policy research. Now, I request Professor Nithashri Mitra Madam to deliver the lecture. May I audible now?
Yes,
May I
ma'am.
audible now?
Yes, ma'am.
So, good afternoon to one and all. First of all, I am grateful to the members of the department for making me part of this memorable day when we all have gathered here to bid farewell to one of our esteemed colleague, Kalpana Garu Madam. Where is Kalpana Garu Madam? Namaskar Madam. It's a very emotional moment for not only for Kalpana Madam but for me as well as other members of the department too. Although we all know that everybody has to be officially superannuated at one point of time, but it is so difficult to let go of a person who has contributed so much for the department wholeheartedly and with whom we spend time as a member of department family. Madam, I have witnessed you and your approach to balancing both professional and personal life, which is truly incredible. You never allowed your personal life to affect your professional life at a workplace. The moment I shared and memories I cherished with you will always be remembered for your kindness and affection. On behalf of not only from me, but on behalf of everyone, I wish you a good luck and your new journey. You would be in our heart forever, and request you to not forget us also. I have a very long association with this department when I was a student, and it was with anthropology. The department, I know that this department has its own legacy, and not only nationally, but also known internationally. In fact, whatever I'm going to present here, the roots of that goes to this department, because I have learned human genetics techniques from this department only. And for that, I am grateful to Professor Gulla Reddy, who allowed me to spend a couple of months here, to Professor V. Raju and Professor J.M. Naidu, who helped me learning in techniques of genetics, and to my dear friend, Professor Paddaya, who always helped me in so many ways. The department has a tradition of togetherness, which I always appreciate and discussed with all others and shared with all others, because I saw that how they are paying respect to former members who has retired or the alumni of the department. For that, actually, Professor Sudhakar, Professor Kalpana, and all other members, Dr. Lakshmi, she is heading the department now. All other members are actually responsible for that. And this tradition will naturally be forwarded a long way, that I know. And I am happy that Madam Kalpana will be definitely going to be meet here again and again whenever I will be present there. With these words, my best wishes are always with you, madam. I will really miss you a lot whenever I visit Visakhapatnam, and if you will be not there, I considered this department or the volunteer as a second home, because I am a very frequent and regular visitor of that place and always feel homely in in the presence of all the members of the department. So what I can say now, I'm also very emotional now. Our well wishes and are always with you, madam, for your good health, wealth, and success in every walk of your future life. So thanks to the department that they have given opportunity to share this occasion with all of you, and I'm grateful for that. Now I'm going to present a very overview what the research we are actually now going, and let me allow to share my PowerPoint, please. Is it visible?
When it is visible, open the slide.
The slide.
You isn't open the slide. Just open it.
No, I have.
We can't see your presentation.
There are multiple.
Yes, ma'am. That is. Yes, sir. Yes, ma'am. You have once open your PPT just once, ma'am. Under the you have minimized the PPT. Just open.
Yes,
Ma'am, once stop the presentation and represent it again.
Okay. Let me try it. There are so many.
Ma'am, once share entire screen. If you go through present, you have 3 options. Press entire screen and highlight.
Entire screen. Yes, entire screen.
Entire screen and highlight the thing and press share option.
PPT.
Yes.
PPT open.
Ma'am, you are still not presenting anything, ma'am. You have not shared the presentation. Means, yes, ma'am. Now it is presenting.
But it is multiple.
Yes, ma'am. Yes, ma'am. Once open the PPT now, ma'am.
Yes, it is open.
Yes, ma'am. Yes, ma'am. Make it full screen. Yes, ma'am. We can see it. Make it full screen. Okay. Okay.
So I am actually giving an overview of the investigation of genetic, physiological, and anthropometric variables in relation to chronic obstructive pulmonary disease in Chhattisgarh, which is commonly known as the COPD. Just I would like to share first the background of this study. As all of you know, air pollution has been consistently increasing in developing countries, attributable to industrialization and traffic congestion, thereby raised the gaseous and particulate matter component concentration of urban ambient air, which is associated with increasing respiratory morbidity. Over recent years, in the public health domain, the PM concentration has become a topic of considerable importance since epidemiological studies have shown that exposure of PM with aerodynamic diameters of less than 10 and 2.5 micrometer induces the increase of lung cancer, morbidity, and cardiopulmonary mortality. You can see the picture of the air pollution here, which is a serious concern for everybody. This is not only the picture of our city, but more or less, you can see this kind of things in every big city in our country, even Delhi and Calcutta, Bombay, etc. So this kind of actually source of this outdoor air pollution and indoor as well as the indoor air pollution is affecting our health because of the PM particulate matter, and the 70% actually is with the household matter, with the outdoor air pollution, and 30% is related to the indoor air pollution. This is the global population, which is indicated here in the left side, and red ink written are the Indian populations. You can see how many actually the cities of India have a very high concentration of the particulate matter. And here only the top 20 cities I have mentioned here, and Raipur. The seventh one is the Raipur, where this study has been conducted. So the key, this is actually taken from the WHO Global Urban Ambient Air Pollution Database, which they made in 2016. So out of 3,000 cities from 103 countries, actually Raipur stood at 144 in number. So the key pollutants of concern are particulate matter, which is mainly sulfur oxide, nitrogen oxide, and the ozone layer. With that, with couple of this, the growth of the population, rural versus urban, is also matters to increase the pollution rate, because more and more people are now migrating towards the from rural area to urban area. So death attributable to these particles, particulate matter, are mentioned in this slide, which I have taken from the published literature given below. So most of the deaths you can see is from the ischemic heart disease, and this is what we have concerned are the chronic obstructive pulmonary disease, or the COPD. So Raipur, which is actually the capital of the Chhattisgarh state, which is a newly actually formed state in 2000, 1st November 2000, it came into existence, is actually an industrially important city with a population of 1.2 million. It is situated between two industrial complexes at a distance of about 30 kilometers, the Bhilai steel plant and the Jamul cement factory. Century cement plant, Larsen and Turbo cement plants, Monet sponge iron, and the power plant Woolworth textile plants are also located in the radius of about 30 kilometers. So this is a very pollutant zone of this, from where we actually have taken the samples. Apart from this, a large number of fertilizer plants, oil extraction plants, agroforest plants, and electrical goods manufacturing plants are also set with this in the circumference of the 30 kilometers. These huge coal burning industries on its western complex produce a large amount of fly ash and PM, which affect the local environment as well as the surroundings by the aerial dispersion processes. Other industry manufacturing electrical goods, iron, steel, aluminium, and copper on its eastern complex. Air analysis of area was done by one of the scientists of our chemistry department, who has reported that the frequency of this particle concentration exceed 150 microgram per meter cube at Raipur, where 100% in all cases, that is in all seasons. Similarly, the frequencies of PM 2.5 concentration exceeding 65 micrograms per meter cube in the same season were also 100%. So all the season, this area, 30 kilometer radius area, is actually have a highest pollution. Their study signifies that the industrial emission in Raipur was high, in all season and played an important role to contribute the PM 2.5 particle loading in the atmosphere. Epidemiological studies have suggested a statistical association between health effect and ambient PM 2.5 and PM1 particle concentration, especially the submicron fraction that can penetrate into the alveolar region of the lungs. It was reported by Deshmukh et al. That these are the scientists from our chemistry department, that PM10 and PM2.5 was most significant polluting agent causing maximum deterioration in the ambient air quality of Raipur. The magnitude of PM air pollution in this region has a significant impact on the surrounding region that implies a higher risk of human health as PM1 and PM2.5 penetrate deeper into the respiratory tract. In addition, owing to the major tobacco production zone in Chhattisgarh, especially in the Raipur also, environmental tobacco smoke exposure, active as well as the passive, is very common that has serious ill respiratory effects on COPD or on human health. In addition to that, there is a national highway, namely Great Eastern Road, that adjoins Kolkata and Mumbai, approximately 400 meters away from the sampling point, with frequent heavy vehicular traffic volume of nearly 25,000 vehicles per day. Analyzed road dust fall and its impact on human health collected from 10 major sampling sites, namely, these are actually we have given the code, that is S1, S2, S3, S4, S5, S6, and S7, S8, and S9 and S10 villages nearby this Bhilai 3 and 25 kilometers west of the capital Raipur, where the AIIMS is also located in the Tatiband area on the main Howrah, Mumbai rail line and National Highway 6. Their results have been shown that lead and lead and zinc concentration is higher compared to prescribed limits in all receptors, and the metal concentration in road dust fall was higher in order of this gradation, that is lead, zinc, copper, then iron, then mercury. Dust particles are deposited in a human body, plants, surface water and soil. They have two potential causes to harm either locally or subsequently elsewhere in environment. Several people of this area are suffering from the dust and heavy metal borne diseases, which is related to lung diseases. Owing to the above mentioned facts and with this background actually, and to fill up the research gap in the COPD research, this actually proposed a research project was undertaken with the AIIMS Raipur with two department members. One is the Department of Physiology and another one that is Department of Pulmonary Medicine. So this industrial zone actually is surrounded in the area of AIIMS. So we have actually thought of collecting sample from this particular zone only. So we have actually hypothesized the people residing near the air polluted area and vicinity of outdoor air polluted area and involved in smoking habit may suffer from respiratory diseases resulting in severe disease condition such as COPD disease. Some of the populations may be genetically predisposed to the disease due to genetic alteration of the candidate genes with reference to COPD. And based on this, the above background, actually, we have taken the objectives to study the anthropometric variables and indices of the COPD patient and the controls, to record the pulmonary function test of the COPD patient and determine the severity of the disease. This part has been done in the AIIMS Physiology Department. To record the resting electrocardiogram of the subjects. This was also done in the AIIMS. To study the genetic polymorphism of selected candidate genes in COPD patients compared to control population, and analyze the statistical correlation of genetic alteration of the above genes, lung function analysis with disease severity, and to find out the correlation, if any, between the time and frequency domain of indices of short-term HRV analysis, heart rate variability analysis computed from the ECG with the blood analysis parameters against the severity of the disease process as compared to normal healthy subject. So little bit about the introduction of this disease. COPD is a chronic respiratory disorder that progresses slowly and characterized by an obstructive ventilated pattern, which is rarely reversible, very often related to tobacco smoking, and that can lead to chronic respiratory failure. Although tobacco smoking is known most important risk factor for the development of COPD, allergic airway inflammation, long-standing asthma, air pollutants, and diesel exhaust particles may also cause irreversible airflow limitations such as COPD. Environmental pollution, age, and airway hyperreactivity are also the risk factors. It is a major but neglected public health problem and is a leading cause of death worldwide and represent a substantial economic and social burden throughout the world. These are the major risk factors for COPD. And some of them actually we have discussed. The outdoor air pollution especially, then the anthropogenic and anthropometric traits also are the risk factors, socio-demographic condition, then the lifestyle variables, tobacco smoking, indoor air pollution, because the most of the sample are coming to this hospital actually from the rural area, and they are actually using the cow dung as well as the wood as a fuel. So indoor air pollution is also a very important source in our samples for this pollution. Then the genetic factors, especially the alpha-1 antitrypsin and the other actually markers are also associated with that. But this is in the initial stage of the project, which we have taken as a pilot project funded by our state government, and now we have actually got this one major project from DST and from ICMR, and we will definitely go for more and more genetic markers, which we have mentioned here, including this. And the occupational exposure of the subjects we have also noted, and review of literature revealed that this is one of the major actually cause of the environmental pollution and the infection. So this is worldwide prevalence of the COPD per 1,000 of the populations, and we can see that males are more affected than the female, and this is the position of the India in the slide. Then this is the research design. We have recruited the subjects. This is a cross-sectional study actually. The subjects who are actually attending the outdoor and indoor of the pulmonary medicine department. We have recruited subjects from the AIIMS of that department as well as from the physiology department. And the case control model we have used here, which is a hospital-based, and the controls are from the population-based, tried to collect the samples from the general population who have not actually any disease or any kind of morbidity, and few of them actually tested for the pulmonary test on the COPD day. So they are negative of that. And the COPD patients as well as the control samples have all again split into the smokers and non-smokers to see the effect of tobacco smoking on this. So in this case control prospective study, actually 100 COPD patients of both genders with age-matched normal healthy control subjects between the age group of 35 to 75 years were recruited from OPD of the Department of Pulmonary Medicine after obtaining a written informed consent of explaining the study objectives and matching of inclusion and exclusion criteria. So 100 COPD patients and 100 age and gender match controls having normal BMI. We have included those samples only who are having the normal BMI, and this criteria, actually to meet this criteria we have to actually reject so many subjects because the patients, most of them are not having the normal BMI. So that's why the sample size we have included here are not much more, although we have collected data more than 250 samples. So after recording pulmonary function test and other socio-demographic and anthropometric data, lead to resting ECG was recorded for short-term HRV analysis, that is time and frequency domain indices, and the biological sample was collected on each subject and further actually underwent as for isolation of DNA and performing the PCR and all these things. And analysis was done using the MS Excel, MS Office Excel as well as the SPSS. So this is the sample size. Although in the physiological variable we have included only 97 persons because we were actually not able to perform the whole analysis. But in other we have this kind of sample size, and most of the sample recruited falls under this category. That is above 60 years of age, and male and female both we have recruited. So this is a anthropometric characteristics of the control and COPD patient. We have taken BMI. For that we have taken height and weight and waist hip ratio. We have calculated using anthropometric measures of standard, following standard methodology, and then tabulated. This is the in the subjects from both the groups were actually included in the BMI range of 19 to 25 only, which is the normal range. A significant higher BMI was seen in the control female as compared to the control male. Though this difference was within the range, normal range of BMI. However, the waist hip ratio was found to be higher in the overall COPD patients when compared to the control subject. The WHO subjects as compared to their counterparts, that is male in both control and the patient, significant at 5.5% and 1% level also. These are the HRV indices and control in control and COPD patients, and this is the graph of that HRV analysis. So the PFT profile, which is shown in this graph. The PFT profile, the COPD group had significant pulmonary function test as expected. So that is FVC, FEV 1 and FEF 25 to 75 and PEFR, and all the PFT profile actually it is found lower in the COPD group, which is this shows the significant difference from the normal one. And this reduction was seen in to be present irrespective of the interesting. This is an interesting result that this reduction was seen to be present irrespective of smoking status and was significantly less in both smoker and non-smoker. When gender difference was evaluated for PFT, control female showed significantly lower value for all the indices from their male counterparts. However, this gender difference was absent in COPD patient except for FVC being significantly lower in female. The time domain measures did not show any difference between male and female, both for control as well as the COPD group, whereas among the frequency domain measures, the control subjects found LF was significantly lower and HF was significantly higher as compared to COPD patient. Also, the LF by HF ratio was significantly higher in COPD group in comparison to controls. Same pattern of significantly lower HF, higher LF and higher LF by HF ratio was consistently observed in both smoker and non-smoker COPD patients. Another consistent observation was that female subjects in control group exhibited significantly higher HF and lower LF and low LF and low LF by HF ratio as compared to their male counterparts. However, this gender difference for the frequency domain measures was not evident in COPD cases except a lower LF by HF in female patients, though it was not significant. So, I'm sorry, one slide was actually inserted inside. So we have analyzed only the two markers, which are that is the GST-T1 and the GST-T1 marker for this COPD patient and control group. The occurrence of GST-T1 deficiency was concluded from the absence of 480BP fragment. Albumin gene fragment used as internal positive control resulted in constant 350 base pair band in all samples for GST-T1 gene individually subject were categorized as having null type, non-null type genotype. The frequency of GST-T1 null significantly differed among the COPD patients and controls. In the smoker and non-smoker group, the difference was non-significant. Genotype distribution of allele frequency of GST-M1 and T1 genes polymorphisms revealed that polymorphisms is in an individual gene may impart only a small relative risk of COPD, and it is likely that the cumulative effect of many polymorphisms will be important in its pathogenesis. So this is the result of the GST as well as the T1 marker gene, and with this I would like to conclude that it may be concluded from our study that changes in the sympathetic and parasympathetic status of COPD patients as depicted by HRV indices with a significantly altered trend is clearly indicative of HRV as an early marker for future cardiovascular risk. Impact of age, WHR ratio and pollution due to very high incidence of COPD in non-smoke patients in our study also emerged as equally important factors in the disease progression and outcome in terms of the cardiac risk. The WHR ratio was found to be significantly higher in overall COPD cases as well as smoker and non-smoker COPD patients. The frequency of GST-T1 null genotype significantly differed among COPD patients and controls. In the smoker and non-smoker groups, the difference was not significant as compared to control subjects. The key message I would like to actually given here is that HRV holds the potential to act as an early marker for the possible cardiac risk in COPD patients, even at the stage when they are free of any associated comorbidity, that is obesity, hypertension, and diversity, etc. So these are few photographs. This is from the COPD day. It is our team from AIIMS as well as from our department, and he's actually the head of the head of the pulmonary medicine department. Now some head of the department has joined, and he's associate professor, Professor Behra, who is also a co-investigator, and Meenakshi Sinha, who is actually the professor of physiology department, who is also a co-investigator. So I am presenting on behalf of them this our preliminary result of the COPD. This is also recruiting the patient in the AIIMS. This is a COPD patient, and this is actually HRV analysis we are doing in the COPD patient. So with this, actually, I would like to acknowledge Chhattisgarh Council of Science and Technology. And actually, it was the request of the government also, because the Raipur is a very high pollutant zone. So from CHIPS, that is the Chhattisgarh Biotech and Infotech Society, of which I was one of the member of that committee. So from that, actually, this topic was emerged, and they have given a very small fund to screen the subject and see that whether what we are hypothesized is true or not, and then go for a big project. So we have got a very good result for that, and we have applied in the ICMR, and about 1 crore we have actually sanctioned. It is just a matter of time that they will release the money, and these 3 departments are actually not 3 departments, 2 AIMS department and Center for Integrated Tribal Studies, which RUSA has been given an innovative center, and I was coordinating the center. So we have taken this project for study of different tribal area also, because this was actually a hospital-based report. So all the machines which we have to take in the field, actually they have procured in the AIMS, and will go very soon. I would like to share here this project actually, because when we are reviewing this, so we find that very few studies have been done on it, and actually it has emerged due to pollution and environmental pollution particularly. It has emerged as a very serious actually disease, and a lot of toll of death has been occurring by this. So the importance of this project I would like to realize to all of you, although it's a very preliminary report, in particularly with regard to the molecular markers. We would like to also I would like to particularly thanks actually the director of All India Institute of Medical Science, Dr. Nitin Nagarkar, who is a cancer specialist and very associated with a very good research work, and always encourage us. And I'm proud to say that this was the first research project which was taken in the AIMS by them, because earlier it was in the establishment of AIMS was going on, and then we approached them, and from so many committees, including from Delhi, it has been passed, and we got the permission to do this kind of research, and now go for a big one. And so his encouragement, enthusiasm, and personal interest actually, because at that time they won't had even the academic council here. So he has taken that kind of pain to pass it from the Delhi, and given me the permission to use their departmental facility also. So I'm presenting, as I told you, that I'm presenting these results on behalf of my co-investigators also. Whenever it will be published, naturally the names of them will be there. So one is the Professor Meenakshi Sinha, who is in the Department of Physiology, and Dr. Ajay Behra, who is in the Department of Pulmonary Medicine, from most of the patients from where we have recruited. And they have also celebrating actually yearly a COPD day. So most of the controls, because it is very difficult to call the controls over in the OPD to test their actually the PFT. So we have recruited general patients, their normal controls, from the subjects who have attended those COPD day during last 3 years actually, to meet the gold criteria of recruitment of the subject. So they are actually PFT, their PFT test was the normal one, and other criteria, which inclusion criteria when they meet, we have included in our. So the collection of data is a very slow process if you recruit the subject meeting all the criteria which has been given by the board. So also I would like to thank all the subjects. That is lot, but not the least, because they have voluntarily and very enthusiastically actually participated. We have prepared a very long schedule of collecting all kinds of data, but they have patiently, sometimes it was not actually completed in one sitting. But they personally, although our team member was there, but they personally also called sometime to us that today or tomorrow we are coming here. So they wanted to give their data very voluntarily and very enthusiastically. That is a very good thing we find in this particular project. With this, actually, I would like to again thank you all that you have given me opportunity to share my small presentation, which once I never forget, and I always actually remember this department who has introduced me in the techniques of the human genetics, and then also the refined it through the Indian Statistical Institute, from where under the leadership of Professor Partha Pratim Majumdar, I have learned so many things, and which actually I think has impact on my career, and I got a conscious. So thanks to the Department of Genetics and all the members of the genetic department. With this, I would again like to say Kalpana Madam and good wishes. Thank you.
Thank you very much, madam, for immediate offer. And again, thank you very much for your words about me. Anyway, retirement is inevitable on everybody who are working. Whether it's state government, central government, teachers, or whatever it may be, madam. Okay, anyway, whenever you visit Visakhapatnam, I will definitely meet you. Even though we are so friendly, our friendship never terminated. Just to remember that one, I will definitely meet you whenever you visit Visakhapatnam. Otherwise, you will visit my home. You can stay at my home. We can spend some time because we are at freedom, madam. We can enjoy our retirement. Thank you very much, madam, once again. Thank you.
Mitashri? Mitashri? Mitashri, thank you very much for obliging our It's always a pleasure to have you for any program in our department. Thank you very much once again.
It's my pleasure to. It's my pleasure. But your voice is echoing from that side.
It's not very audible or what?
It's audible but not clear. Echo is there.
Oh, okay. There is some problem with the mic. But okay, you understood what I said.
Yeah. Okay. It's always pleasure.
Hitachi Mitra has delivered a beautiful speech regards to our respect and connection with our department, our association with Dr. Lakshmi Ratnam. I thank her for accepting our invitation to me. Madam has exposed an elaborated lecture on investigation of genetic, physiological, and anatomical variables in relation to chronic obstructive pulmonary disease. He has listed what are the risk factors of COPD, and she has also discussed about the significant results that came out of our study. Thank you once again, Madam, for accepting our invitation. So before the valedictory speech by our respected registrar, Professor V. Vishnu, we have to first summarize today's sessions, what happened and all, then Sir will join to deliver his valedictory speech. Morning, we started with the inaugural function at 9:30 a.m. and in the absence of our Vice Chancellor, we invited Dr. P. Shyam Prasad Garu, Honorable Vice Chancellor of India Health University, as the Chief Guest for today's inaugural function. And Professor K. Srinivasarao, Principal, College of Science and Technology, Andhra University. He has raised the occasion and gave a message to the participants. Then, followed by that, we conducted 3 academic sessions. And the first speaker was Dr. Venkata Ratnam, who is Associate Director, CFC Scientists, Postal CFC, who is the prestigious alumnus of this department. So Dr. Venkata Ratnam, in his lecture, in the first half of the lecture, he started from what gene is and the history of gene therapy, the evolution of gene therapy, what are the different types of gene therapies, and he also touched upon the concept of gene editing using CRISPR-Cas9 technology. He also discussed about cell therapy, the opportunities and challenges in that kind of therapy. And in the second half, he discussed about the gene therapy that is carried out on some retinal disorders in his company, what they are doing. Then the second speaker was Dr. Raghunath Rao, who is a renowned oncologist and president of the Academy of Sciences. The sir discussed about the role of genetics in precision diagnosis and personalized treatment. He discussed about the treatment that was available for cancer 2 or 3 decades back, when the treatment used to be indiscriminate. That is, the cancer cells cannot be distinguished from the normal cells. Treatment was common. But now we are in a stage that we are targeting the cancer cells exclusively. And the diagnosis and treatment of cancer changed a lot of transformation took place, and that was with the help of genetics. Only with the help of genetics it was possible for precise diagnosis, not only for diagnosis, for treatment also. We are using now the genetic methods. And he also described about the different kinds of mutations that were observed in different cancers like melanoma, acute leukemia, and all. Classification of cancers, which earlier it was used to be done based on the histological pattern. But now the cancers are classified based on the genetic economy. That's what we discussed. Then we also discussed about the different types of drugs that are used for cancer treatment. And these drugs are helping in silencing some of the gene expression. So by silencing the unwanted gene expression, we can treat cancer. Discussed about different kinds of immunotherapies that are available for treating cancer. And the different ways in which the cancer patients they respond to drugs based on their genetic makeup. Finally, he concluded that an integrated approach is needed for the enhancement of survival rates. He also discussed about stem cell transplants to treat cancer. So there was a tremendous response for both the academic sessions, and in the afternoon, the third speaker was Professor Geetashri Mitra. She's a retired professor from Pandit Ravishankar Shukla University, Raipur, Chhattisgarh. Madam is very closely associated with this department, and she is very happy to participate in this webinar to give a message on the occasion of the superannuation of Dr. Lakshmi. She is very much associated with Madam. And she discussed upon the chronic obstructive pulmonary disease and the risk factors of COPD. She highlighted this based on the pollution, which is very increasing tremendously nowadays, and this has a very high influence of COPD. So she discussed about the work she has done of COPD patients in Raipur, and she has she gave the important observations that she made in her study. She also found a marker which has a risk for cardiac problems in COPD. She also found some markers which are showing like GST1 null, which is significantly different in cases when compared to the controls. So she did some anthropological studies and genetic studies also. With that, we completed the 3 academic sessions in today's webinar, and I feel that each and every session was invaluable, and these sessions have provided insight into different kinds of diseases, different kinds of therapies that are available, like the latest in cell therapy and gene therapy, what Dr. Venkataratan has described. So, I hope that all our students benefited from today's webinar. So, before our respected registrar gives the valedictory speech, we need to take feedback from at least some participants. regarding this webinar. So first I request the students who are present here in the seminar hall of Human Genetics Department regarding their opinion about this academic sessions conducted in this webinar. Yes.
Very good afternoon to everyone. First of all, very thankful to Rohit for the department for organizing this type of seminars, because these seminars are very valuable for us. And in this webinar and about the recent advances, we got to know what are the recent advances that have been done for the prevention of the genetic diseases and the use of genetics in the studies, in all the studies. I mean, in each and every subject, genetics is included. I mean, for any new disease, there will be a genetic cause, because knowing of the genetics and how to treat that with genetics, it will be very easy to prevent and cure for that disease. And in this webinar, in the recent advances, we got to know how to use the nanotechnology and how many gene therapies are there. So, we just gain knowledge about all that things. And the most disease is at present is cancer. So we got to know how cancer can be treated and first of all, how to identify the cancer. I mean, in previous years, as all others said, one cannot differentiate the cancer cells. But after that, as the studies are improved, one can differentiate the cancer cells and how to prevent the cancer cells. And the recent advances using viral vectors, how to cure the cancer cells, and using nanotechnology, using anti-angiogenesis techniques, and all that. We got an idea about different aspects, and that is an advance. Thank you. Respected professors, the guests, the special speakers, postdocs, fellows, all my adults dear friends. Today I will share a particular student of human genetics, take this opportunity to thank our department and along with that all the guests who gave us a valuable lecture on various topics. Being a student of human genetics, it is not new to us to hear all those terms. Nanotechnology, cancer, the therapies, we are well aware of that. However, getting to know much into depth is something that made us all very happy. It was not only interesting, but it grabbed our attention and it made us think that yes, these are the scopes that will be available. There will be further more advances that will take place, be it cancer or the gene therapy or the sequencing which is used for gene cloning and all this. Whatever lectures was delivered to us, we came to know that with the advent of time and with the advent of technology, things are definitely proceeding. However, the disease is also getting complicated. As our respected Kadriyas are told, that most of our cancers because of our own health, our own lifestyle and all. So it is indeed important for all of us to take care of ourselves because our health lies in our own hand. And as a genetics student, let me tell this also that genetics is not just genes, it is a combination of heredity and environment. So the environment definitely has some more impact on us. With this webinar, whatever it was conducted. It is indeed prestigious as getting to know about recent advances, recent technologies from the esteemed professors, the scientists and the doctors itself. We thank all of them because as a student we are really fortunate enough to be a participant of this webinar, which was conducted even though it was online, but with a combination of both online and offline mode. And I would just end by saying that being a student of Andhra University is itself prestigious in itself, because this is the first department, the first university that started the course of human genetics. So thank you very much. Thank you, professors, and thank you all the guests for giving us your valuable lecture and taking out time for us. Thank you.
Welcome to our respected registrar, Professor Vishnu Mohan. Sir, we have completed all the 3 academic sessions which we scheduled today. We have summarized all the 3 sessions and took feedback from the students, and we are waiting for you to address the participants and give your guidance.
Madam, am I audible?
Yes, sir. Yes, sir. You are audible, sir.
Okay. I see Vice Chancellor, NTR Health University. Namaste, sir. Dr. Shyam Prasad. And nice to see the delegates and also the webinar organizers. And as I understand, that the webinar on recent advances in human genetics is a
meeting.
ఇక్కడ కూర్చోబెట్టలేదు 10 నిమిషాలు. Madam, am I audible?
Yes, sir. Yes, sir. You are audible, sir.
Okay. Because the Vice Chancellor NTR Health University, whose conversation is coming on phone, therefore I stopped a while. So I am so glad that the webinar on recent advances in human genetics is going to end now, but this is not an end, this is only a beginning. Whatever you have learned through this webinar, right from morning till now, as I understand through feedback, those who are actively involved in the learning process, they have taken the inputs in right direction. Definitely, the recent advances in human genetics will go a long way in undertaking research in those directions that this discipline demands. See, normally in any discipline, recent advances definitely are going to be a great concern for researchers, and those who joined in this webinar undoubtedly at the end benefited with those points, with the consensus of all those resource persons, people who interacted post presentations with the resource persons might have definitely benefited a lot to all those whoever has registered and also attended with all that sincerity to know what are the recent advances is one aspect in human genetics, how they are going to be the future directions, and what can be taken up by the future researchers in these areas. See, as I understand, as a layman in the subject of human genetics, the modern human genetics began when new advances in genetics were systematically applied in medicine right from the year 1949. This is what I have picked up from the note. And all of us should believe that there is a close relationship between the genetics and medicine evolved into human genetics. So there are two specific branches. One is genetics and medicines. When they are combined, they evolved into the discipline human genetics, contributing greatly to an understanding of the causes of human diseases at root level. So it has evolved in different directions, mainly based on, of course, different methods of investigation. Methods of investigation are never same. Always there is divergent approach in order to reach at convergence. This is order of the day in research. Unless there is a convergent ideas which can be pooled and discussed, debated in order to make them converge at one particular point of time with a consensus. Always the consensus arrived at is a superior view, acceptable view in the scientific world. If you take what is happening in the field of human genetics, today the human genetics comprises of genomics, molecular genetics, tumor genetics, pharmacogenetics, immunogenetics, right? Then epigenetics, cytogenetics, somatic cell genetics, biomedical genetics, population genetics, bioinformatics, and many other emerging areas which are arriving today. Probably this is the reason why today's webinar on recent advances in human genetics is really a noteworthy, contemporary, highly relevant in the field of human genetics students and researchers and faculty. So the dual structure of being both a medical and a biological discipline makes human genetics unique among the medical and subspecialties. If you look at applications, as I understand, the field of cancer genomics is a relatively new research area that takes advantage of recent technological advances in order to study human genetics at a bigger space. Cancer genomics research definitely contributes to precision medicine by defining the cancer types and subtypes based on their genetics, providing patients with a more precise diagnosis and treatment. The targeted therapies specifically with a view to combat characteristics of cancer cells, making the patient to less likely to a likely toxic state when compared to the chemotherapy, radiation, which are widely prevalent till recently. The unprecedented levels of control over nucleic acid delivery, modulation of immune system, and precise manipulation of human genome technologies are not imaginable 10 years ago. Recent success in genetic medicine have paved the path for a broader second wave of therapies and laid the foundation for a next generation technology. Right now, gene therapy is at an inflection point. One of the greatest challenges in the field of cell and gene therapy in near future will be in the area of regulatory sciences, accommodating the unique challenges posed by the innovative challenges and technologies as we proceed further to very personalized therapies. Now, one such invention in the recent past is stem cell therapies. This is how today human genetics is truly a multidiscipline and touching every part of medicine. And unless we view at different dimensions and make all these views together, with precision, both diagnostic and treatment can never be made possible. So whatever challenges that we come across today, like decoding the cells and suggesting proactive medicine, and also customizing medicine to suit to the genetics of a patient or an individual is going to be the order of the day. Therefore, the topic on which this webinar is organized on recent advances in human genetics, as I understand, has definitely contributed to the researchers and students in a meaningful manner, and whatever points they have taken today will go a long way in building their professional career, future research, improvement in technology usage, and also at the end, proactive health management.
Is it over, madam?
Just a minute.
Hello?
Sir, you are audible. Sir, you are audible. You are audible.
Disturbance. అయిపోయింది కదా చెప్పండి.
Sir, you are audible.
Yeah, shall I start again or you heard at least some part of my message?
Just it's a part of message heard.
See, again, sorry for the interruption. These are all, technical difficulties. What I am explaining, a webinar on recent advances in human genetics, comprising several branches like genomics, molecular genetics, tumor genetics, pharmacogenetics, immunogenetics, epigenetics, cytogenetics, somatic cell genetics, biochemical genetics, population genetics, bioinformatics, and many other emerging areas. It is all because of the dual structure of being both medical and a biological discipline makes today the human genetics subject is so important. Am I audible?
Yes, sir. Yes, sir.
All others, please cooperate and keep your mic under mute. See what I said, the dual structure of being both a medical and a biological discipline today makes human genetics unique among the medical subspecialties.
See, if you take the field of cancer genomics, is relatively a new research area that takes advantage of recent technological advances to study human genome. Cancer genomics research contributes to a precision medicine by defining the cancer types and subtypes based on their genetics, providing patients with a more precise diagnosis and treatment. See, the targeted therapies specifically combat characteristics of cancer cells, making the patient to less likely toxic state when compared to the age-old practices of chemotherapy and radiation. See, the unprecedented levels of control over nuclear and acid delivery, modulation of immune system, and precise manipulation of human genome technologies are not imaginable 10 years ago. The recent successes in genetic medicine have paved the path for a broader second wave of therapies and laid the foundation for next generation technologies. Right now, gene therapy is at inflection point. One of the greatest challenges and changes in the field of cell and gene therapy in the near future will be in the area of regulatory sciences accommodating the unique challenges posed by innovative technologies as we proceed further to very personalized therapies. This is how in the recent past we heard about stem cell therapies, how the chronic diseases can also be addressed with the stem cell research and therapies. This is how human genetics is truly a multidisciplinary subject, and a webinar on recent advances in human genetics might have used experts in their own domains from different areas which are connected from all over the world, so that the research activity undertaken in each of these sub-branches, domains, they are going to enhance the ability of understanding problem from multidimensional thinking. As all of us know, always the research begins from ambiguity to specificity. Why research begins with ambiguity is due to different approaches, different methodologies, different technologies which can be addressed to a given problem. A webinar of this kind, definitely with the knowledge on recent advances in genetics from different fields of study connected to human genetics, definitely would bring ideas, divergent ideas, which can be debated, discussed, and finally reach these divergent opinions, views, which are exchanged, and to arrive at clarity of thinking, eventually, as I understand, will lead to convergence. This is what is required in a research. From divergent thinking to convergence, if you are able to reach, definitely you find a solution. The solution that emerges in human genetics is not simple. Definitely it is a complex. When you engage people with different backgrounds, with varying capacities and competencies in these disciplines, when they address problem from a holistic perspective of all these dimensions, definitely from divergent thinking, when you reach the convergence, there is a solution with all that creativity. When these creative minds who are specialists and super specialists and micro specialists, when they do research, identify a breakthrough Definitely all such breakthroughs, when they are combined together, there is a holistic perspective and a practical solution to a defined problem. From that perspective, this today's webinar, as I believe through feedback and messages that are splashing on my screen, people are so happy that they get whatever they wanted by joining as delegate with the excellent resource persons. Whoever has delivered resource lectures on this topic of recent advances in human genetics definitely would build the competencies among the students, researchers, and the faculty in deciding the scope of future research and taking research to a step forward in making all efforts when combined. A complex problem can be made simple, and a solution to that problem can be evolved because of the involvement of people from different disciplines. This is what we teach in management subject. Very similar. Human genetics is also a multidisciplinary subject. Webinars of this kind definitely would identify recent advances in their own fields of specialization, which we call domains of research. And such research, when they exchange, would facilitate people, whoever is interested in either teaching, research, career in the field of human genetics, towards the right path, towards taking the ideas, more realistic problems and creative solutions. And customization can be done to the patients in suggesting medicine. If you address at root level, find out the causes through bioinformatics and genetic engineering, we are able to solve chronic diseases. That is going to be the contribution of this one-day seminar, one-day webinar on learning the recent advances in the field of human genetics. And I see Vice Chancellor of NTR Health University, Dr. Shyam Prasad Garu. Probably the delegates, whoever has registered, the scholars, the faculty, definitely must have benefited with the presence of learned personalities, experienced professors, the veterans in the field of research. When we discuss these things, all of you think that, through this, any bigger problem can be addressed, and also information can be exchanged in defining problem with precision. This is one aspect. And the other aspect is, through this webinar, probably you are already networked. The ability of networking in completing bigger tasks by the researchers and faculty will go a long way in building collaborative research, joint research, and also building relationship for future benefit. And also, of course, the present research can also be improved through these interactions, through discussions. When there is a common platform provided in this webinar on recent advances in human genetics, definitely, might have created, worthy information, valuable information, and immense value to all those, whoever has taken this webinar with all that sincerity. Definitely need to commit. The future research should be in the right direction, so that India is going to become a far ahead in genetic engineering and human genetics, so that the diseases can be addressed with all that confidence in us. And in the years to come, without medicine, probably through genetic manipulation, long-term diseases, chronic diseases, illness can also be treated. And the future research should be in the direction that is going to make practicality in the research and connect to the real problems of the mankind living on the earth. Today we are living in a very pandemic situation with a fear in our mind that there could be a third wave, fourth wave, so on. Every day we hear a lot about new viruses, new problems that are coming up, and new diseases that are going to come up. So the more research that you undertake with all that intensity in your mind, with complete understanding of all these dynamic forces which are operating on the health dimension of mankind on the earth, definitely would build, the future research, simple solution to your biggest challenge and biggest problems. I take the opportunity of really congratulating the organizers, the seminar director, co-director, and the faculty, whoever is involved in organizing this one-day webinar, so meaningfully, usefully, and addressed the most challenging dimension of recent advances in human genetics, creating awareness, interest in those areas, so that the future research is going to become really in the right direction. That is what is the output of a webinar should be, apart from networking, as I said earlier. And I really congratulate the organizers, profusely thank the resource persons, whoever has enlightened and shared their knowledge, expertise with the delegates, and also the delegates, whoever has registered for continuation of their thinking process in those dimensions of recent advances, so that the future research is going to be more meaningful. I thank everyone, whoever has given me this opportunity to share some of my thoughts, and also see our well-wisher, Vice Chancellor, Dr. NTR Health University. Once again, thank you, sir. Thank you very much for your gracious presence, and also all other resource persons. On behalf of the organizers, I am very thankful to all those who made this webinar highly productive and successful at the end of this day. I feel proud that I am invited at the valedictory session to associate. Thank you all. Wish you best of luck. Over to organizers. Sudhakar?
Sir.
Lakshmi garu?
Sir.
Yeah.
Sir. Is it clear?
My job is over?
Over, sir. Over, sir. But
Okay.
make a vote of thanks, Head garu. 1 minute.
Okay.
1 minute. Only 1 minute.
Thank you very much, sir. Thank you very much, sir, for your valedictory address. Sir, we know that you are in a very important meeting and you have taken your time to give the valedictory address to the participants of this webinar. So, sir has explained the multidisciplinary nature of genetics and the importance of genetics in precision medicine. Sir, you have also explained us the significance of conducting this kind of webinars. The use of conducting these webinars is mainly two. One is to build the networks, and the second one is to try to get some solutions to the existing problems like treatment of diseases and all. Thank you very much, sir. Now I take it as my honor and most privilege to propose all vote of thanks for the one-day presentation webinar on recent advances in human genetics, RAG 2021. First of all, I thank our beloved Honorable Vice Chancellor, Professor P. Prasad Reddy, for his immediate acceptance and encouragement for organizing the webinar. I thank the Chief Guest, Dr. P. Shyam Prasad Garu, Honorable Vice Chancellor, Dr. NTR University of Health Sciences, Vijayawada, for his valuable inaugural speech. I thank Professor K. Samantha, respected Rector Madam, for agreeing to be a factor. I especially thank our beloved Principal, Professor K. Srinivas Rao Garu, for personally gracing the inaugural function. I extend my profound gratitude to our respected Registrar, Professor B. Krishna Mohan Garu, for his invaluable support and for the wonderful closing remarks in the valedictory function. I thank our teachers, Professor G. Ballareddy Garu, Professor P. Deeras Garu, Professor M. Satyanarayana Garu, Professor P. Venkateshwara Garu, and Professor V. Krishna Rao Garu for their blessings and constant encouragement in all the initiatives taken in the department. I thank Professor Venkata Ratnara, Associate Director, Students and Scientists, USA, who is the illustrious alumnus of this department, for his vast lecture of advances in cell and gene therapeutics. I also thank Dr. Raghunatha Rao, renowned oncologist, for his detailed explanation on precision medicine and how this therapy will help in enhancing the survival rate of cancer patients. I thank Professor Hitashri Mitra from Pandit Ravishankar Shukla University, Raipur, who delivered a talk on investigation of genetic, physiological, and anthropometric variables in relation to COPD. I thank our Honorable Professor G. Fataya Garu for his active participation and support in conducting this webinar. I am grateful to Professor G. Sudhakar and Dr. P. Ashwini Hasana for their priceless support in every step of this webinar. I also thank the organizing committee, members Dr. Uday Kumar, Dr. Mahankali, Dr. Sridevi, Dr. Rajeshwari, Dr. Lakshmi, Dr. Sudhakar, and Dr. Vidya for their immense help in organizing this webinar. Special thanks to research scholar Monica Sindhu for her innovative designing of the webinar material within a short span of time. I also thank Mr. Teja, research fellow from Biotechnology Department, his help in getting the webinar material in time. I thank the students of the sister departments of Advanced Sciences Bhavan for their valuable participation. Special thanks goes to non-teaching staff of Advanced Sciences Bhavan for extending their meticulous efforts in organizing this webinar. I am extremely thankful to Professor Ramasudha Madam, Honorary Director, Computer Center, and her team for the technical support in conducting this webinar. I also extend my thanks to present media for the coverage. Last but not least, I thank the students of human genetics for their enthusiastic and energetic participation both in organizing and attending the webinar. Thank you. Now I request the audience in the seminar to please stand up for donation.
It's a privilege. Let's all join with it. Please say it.
पंजाब सिंधु गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मागे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जय हे।
Madam, goodbye.
Done.